I'll be here again. Thanks so much.
What you're looking at are some steel sculptures that we collaborated on. We built these structures as representations of data from Klaus Schulten's lab at the Beckman Institute. The data was the folding of a protein called vilin, and the protein folds in 7 microseconds. So, I mean, it's thousands and thousands of times faster than the blink of an eye. And what we did was we took 5 snapshots along that trajectory, and with those coordinates, fed it into a program that would give us cutting diagrams for steel. And then really over the course of a month, students got to learn about protein folding, some pretty difficult biochemical research problems, while at the same time welding and grinding and creating the art.
These sculptures show wonderfully how this protein is first very elongated, artificially elongated actually, and how it then coils up into the characteristic shape that the protein assumes.
Proteins are amazingly beautiful structures. They make us run, they're everything. It's like the basic blocks of what we call being alive. They have a beauty of alien technology to me. It's a world that is perfect, and I'm amazed by how little we know visually about that kind of world.
In the domain of living cells, which is at the molecular domain, this size, scale, it is just difficult for our microscopes to visualize things. With light microscopes, you don't have the resolution, and electron microscopes are not really well suited for studying the processes in living cells.
When a protein is produced, you can kind of think of it as starting out as this floppy string of beads, and then it folds into a final structure. The string of beads, each bead is an amino acid. There's 20 of these things. And the amazing thing about proteins is that they perform all kinds of different tasks. And the task that they perform is dependent on the shape that they take. And the shape that they take is dependent on the sequence of beads.
The proteins in the cell have a remarkable ability to find their proper form. This is very essential for them carrying out the proper function. And it is also very essential for avoiding pathological circumstances like Alzheimer and other central nervous system diseases often come about from the proteins to actually misfold, not to assume the proper form.
And really one of the great unsolved problems in the area of biochemistry is the protein folding problem. That's what one of the things the Schulten's lab is working on, right? Can we, with a computer, watch a protein fold trillionth of a second by trillionth of a second, and actually from that get a movie of what it might look like as that structure forms?
I built over the years what I like to call a computational microscope. That is a microscope not made of glass lenses and metal tubes, but rather made of concepts and ideas from physics and chemistry and mathematics, and made of software and computers that put those ideas and concepts together to simulate what is happening in the cell. Vilin headpiece is a particularly small protein that folds also fast enough that the computer can just manage to follow its birth from being a stretched out, completely nascent protein to a protein that is then functional after the vilin have folded itself into the proper form.
It's one of the proteins, one of the few proteins that with the state of our computers today, we can accurately predict its folding trajectory from unfolded to folded all the way, right? For proteins that are slower than vilin, we don't have the computing power to do it. And I wanted the whole story.
So I published the work and I told the world about it in many lectures. And so it happened that some artists, scientists became aware of it.
Klaus Schulten's work is I know it really well because I'm always interested in seeing what's beyond the static aspect of a protein structure. For the last decades, people saw only the static aspect. They see only a protein as something unmovable. But it turns out the moving aspect of the dynamics of a protein is absolutely crucial. And his group does an amazing job in visualizing that.
I was out there looking for resources that would help me teach concepts of protein folding. In particular, I was looking for something that showed motion. And these supplementary videos that they published really struck me as being very powerful for communicating the idea that there are multiple paths to a single native state. You could see these proteins getting stuck in local minima and getting out of it, and you could visually, you could watch these things go through different paths to the same final structure.
Many people who see for the first time what we actually offer scientifically, they think, oh, we are just using beautiful colors and Disney-type movies. But that is true in a way, but it's also much more than that, because this is the only way actually of grasping with the human mind the kind of complexity that is natural to the living cell.
What I really love to do is to walk around these things up there. You look at them from one direction and you see a timeline. You see that protein folding. You go to the other end, you see it unfolding. And as you walk around, you get a better sense, I think, than you would from a two-dimensional still of the space that helices take up, of the proximity of one backbone to the next. You get a better picture of a protein.
It certainly provokes people to ask, and if they hear the answer that this is actually representing something and it's not a modernist abstract sculpture, most people are actually quite intrigued by that and start asking more questions and wonder about their own bodies.
It's way beyond making pretty sculptures for the public and getting people to think that science is cool. It's way beyond that. You can see things through art that maybe you would have let slip past.
When you see that object under the microscope, you are impressed with the static beauty, but when you see a cell, you are impressed with the liveliness of it. You see things streaming and moving and bubbling and sculpting, and so that is life, and that's what we want to show the world. And art is an absolutely natural way of bringing about the message.
We are ready to begin our afternoon session. Our first speaker will be Axel Brunger. Some of you know Axel for his work with the Export program. So I heard from Klaus that Axel is one of the fastest students of PhD in less than 2 years, and also one of the first. We are looking forward to it. James Gumbart, also known as JC. So JC and I overlapped for 1 year in Boston Lab. So JC got a degree from our University of Physics department. He also worked in the system architecture. And then he went to Argo National Laboratory where he worked with Remodeling. And he became assistant professor at the Georgia Department of Physics in 2013. And we are looking forward to the lecture.
I think this would be fun. Thanks. So it's really a great honor to be here, particularly to be scheduled between two great leaders in the field, Axel and then Rommie after me. So today I'm going to tell you a little bit about really both my scientific work and a little bit of my personal history and how I came to that and how I'm continuing to work in this area of membrane protein insertion. Ahmad stole my thunder a little bit showing this picture yesterday, but you may not have noticed, it was a bit small on his slide, that Klaus is on the wrong end of the cannon. Nonetheless, he wasn't afraid, and I think that's a lesson many of us took from him, to really be bold and not afraid in pursuing our goals. So, I don't really believe that success itself in one's life hinges on a few decisions or one decision, but certainly the path that one's life takes hinges upon, chance encounters and key decisions. And so when I was a first-year grad student here at the University of Illinois, all the way back in 2003, 2004, I had to decide what I wanted to do for my PhD. And so, like some of you have already mentioned, like Axel and Alek, I didn't know how to spell biophysics either at that time. And but I had seen Klaus present to the first-year grad students, and his enthusiasm was really infectious. And so, shortly after I saw this, I reached out to him and I said, "Hey, I'm some no-name first-year grad student. I was wondering if I could meet with you." And so this was late at night, and so I'm thinking, okay, I'll get a response hopefully the next day. But no, within 3 hours, he responded to me saying he would be happy to meet with me. And so I also, unlike some people, I didn't really have an appreciation for who Klaus was at that time. And so I went to this meeting with him, and after only 15 minutes, he decided to take me in his lab for the summer. And to this day, I still don't know exactly what he saw in me in those 15 minutes, but I'm very thankful for that. So I started in the summer of 2004 in the lab, like many physics students, after I finished my courses. And after looking at a few different projects, ultimately I landed on one on a protein conducting channel called SEC-Y, or Sec61 translocon. And so the crystal structure had just come out a few months before I joined the lab, so it was very hot. And in fact, I came to learn that Marcos Sotomayor, my office mate, had looked at it first, but he was too busy with other projects, so fortunately I got a chance at it. And so I just want to introduce a little bit about this. So this channel, you see here, it's a membrane channel. And what is particularly interesting about it is it provides a pathway both for proteins, unfolded proteins to cross the membrane, in the case of, say, secreted proteins, but it also is a pathway for membrane proteins to get inserted in the membrane. And so one of the most common ways this occurs, and certainly the only way that's conserved in all organisms, is called co-translational translocation, in which the ribosome, when it's synthesizing a protein that has this secretion signal sequence, gets targeted to the channel, SEC-Y, or Sec61, where it then binds, and then continues its translation while at the same time inserting this protein into the channel, from where it can either go into the membrane or across the channel to the other side. So I was just a few months working on this project, still in 2004. In the fall, I had just passed my quals, thankfully. And so I was given another great opportunity to attend a workshop as a teaching assistant, actually. So this was only the 4th workshop at the time, so they were still relatively novel. And so we were going to Boston. This was in December of 2004. So I was there, Klaus, Marcos, Zan, Patrick in Zan's lab was also there, and a number of eager students at the time. And so when I realized I was going to be going to Boston for this workshop, it occurred to me that, and I brought it up to Klaus, that, Tom Rappaport, the person that crystallized this protein, SEC-Y, happens to also be in Boston at Harvard Medical School. And so Klaus said, well, let's email him, let's go meet him. And so we reached out to him, and Tom graciously agreed to meet with us on a Sunday afternoon, no less. and we could present some of our simulation, our early simulation work to them. So I save everything, so I still have these slides, almost a bit embarrassing now. So this is just a little bit, or actually most of what I showed to Tom. I just had a few slides that day. And so there was some very, very boring details that probably didn't mean much to him about how the system is set up, how we equilibrated it. I had a nice picture, what I thought was a nice picture at the time. Of course, lipids obscuring the protein, that's a no-no. and then I showed a little bit of some steered MD simulations that we had done. And again, some boring details, but if you're quick with the unit conversion, you might realize that this was insane. This was 50 angstroms per nanosecond that I was pulling this through the channel because I wanted to see something happen in a day of simulation. Of course, even at that time, that's really way too fast. I was very lucky, though, that Klaus didn't fire me on the spot. He actually was very supportive and, particularly in this meeting, very kind to me and talking me up to Tom. So even from this relatively inauspicious start, actually this project turned out very well. Kind of jumping to the end here, Klaus and I have 9 papers together on the translocon sec Y in various forms, looking at various aspects, and also including, along the beginning it was just us, blazing a trail on this project. Eventually others saw fit to join us. So for example, Elizabeth and Leo joined me for a publication. We also worked on a couple of great papers with Chris Chapeau looking at free energy calculations, and even at the end, really a capstone on this project, working with Benoit. And so in fact, this has even continued a little bit beyond, and so although none of these were with Tom Rappaport at the time, more recently I have published a couple of times with Tom now. So I want to focus, though, that's a lot of work there, but I want to focus on the aspect of membrane protein insertion that I mentioned in the title. So sec Y, so looking here from top down in these images, it has a lateral gate that can open to allow the passage of membrane proteins, roughly, one transmembrane segment at a time. And as I mentioned before, this is often done, not always, but often done co-translationally. So you have to have a ribosome bound to the channel while it's translating, inserting the membrane protein, which then, through some various mechanisms, goes into the membrane. So it shouldn't come as a surprise to many of you that Klaus has long been fascinated by the ribosome, and so this was a natural target for us to try to understand how they come together and interact. But at the time we were working on this, so this was around, we started looking at the ribosome translocon complex around 2007 or so. The best structural data, and actually, in fact, to this day, the best structural data on these complexes come from cryo-electron microscopy. And so at that time, in fact, in 2007, a new cryo-EM map of a ribosome translocon complex was released, or was published by Chris Dickey at Boston University. So Chris was also a longtime collaborator of Tom's, Tom Rappaport. And the timing of this was quite fortuitous because we have this great new map, and around the same time, thanks to many people in the lab, like Elizabeth and Leo and Alek, we also had a great new method called MDFF, Molecular Dynamics Flexible Fitting. So I don't want to go into too much detail here, but the basic idea is that the cryo-EM map can be used as a guide for atomic structures in simulation to make them, turn them into a conformation that conforms to the map. Okay, but obviously you need 2 ingredients to this. You need crystal structures to start with, so thankfully we had crystal structures of the ribosome and of the translocon separately. And you also need a cryo-EM map. And so we had to convince Chris Akey to give us the map. Actually, it wasn't, I don't know if even now it's always necessarily the case that when a map is published that it's released. And so Chris was still holding on to it. We had reached out to him, and he was a little reticent to give it up right away. And so Klaus, in his style, what was clearly becoming a pattern, said, well, why don't you go to Boston and convince him? And so, sure enough, so I went to Boston, and basically, I prearranged it to some degree, but met with Chris Akey and convinced him to give us his map prior to its release on the EM database. And in fact, just maybe a brief aside, one of the things I still remember that Chris told me that may have made the difference was that he said in his teaching he had found on the web many of Klaus's movies and images and had found them incredibly useful. So that was, may have played a role, not even realizing it. So, okay, so we got the map, we have the structures, we have this new method, MDFF, so we're getting somewhere, okay. But there was some added pressure that came along. This was Biophysical Society meeting 2008. So Benoit might remember he was one of the organizers of that meeting in Long Beach. And so I don't know if it was explicitly for that reason, but at that same meeting, Klaus was invited to give a symposium on the translocon sec Y. Okay, so sounds great. We're going to get our work in a high-profile venue, seen by a lot of people. But I say it's a blessing and a curse because so on January 1st of 2008, I get this email from Klaus. So it starts out very nice. Happy New Year. Oh yeah, thanks. My advisor sent me Happy New Year. I'm horrified about the upcoming lecture on our translocon work. We need to get it done, to have a strong section on the lecture showing the behavior of this system. Please get started as soon as possible. The lecture will require a lot of work and a lot of luck this month. And so began one of the worst and hardest months of my life. But it was all worth it. So it all paid off in the end. The lecture, I think, went over quite well. We did have some initial results to show, some nice movies. And ultimately, we published this work. So I'm showing here, you can see the faint outline of the cryo-EM map and our fitted structures of the ribosome translocon complex. And for example, highlighting some of the interactions that we were able to resolve at an atomistic level thanks to this work. And so we published that in 2009 ultimately. Okay, so around the same time, around 2008, summer of 2008 in fact, so Klaus and Zan happened to meet Roland Beckman in Munich. So he's at the LMU there, and he was enthusiastic about collaborating actually on a few projects, but in particular, Klaus found out that he had a new map of a ribosome translocon complex, and this one was even more intriguing than the previous one, in this map, so this is the ribosome inside, attached to sec Y inside a nanodisk, it was in the act of inserting a protein. So, okay, so for the first time, we're seeing this in action. Okay. So it shouldn't come as a surprise anymore that the next thing I knew, I was on my way to Munich to meet with Roland. And so actually this was one of a few times I went to Munich to work with Roland. I was very, very fortunate, collaboration. And I was also very lucky that Elizabeth hosted me actually on a few of those trips. So it worked out really well. And so again, so we were able to get the map. We had this great method, MDFF. And so this was the end result. So going from the map to the fitted structure, we could really see, so this is the ribosome up here, the translocon in a membrane, and in green you can see the inserting nascent protein, nascent membrane protein. Okay. And so this was the first time we'd been able to see this at atomistic resolution. And one of the things that was really nice about it was that it confirmed a lot of earlier ideas about how this channel worked and about how membrane proteins get made. And so, in fact, if you're not convinced by me. So you can also tell how important it was by how serious we look in this picture. All right. So that's just a brief, very brief look at our translocon work together. But it just so happens that, so in 2006, I had another chance discussion, this time with Ahmad in the lab. I think we were chatting in the TCBG library, and he brought to my attention another interesting membrane protein that he had just learned about, called B2B. So this protein is a gram-negative outer membrane protein that's responsible for the import of vitamin B12 into bacteria. So the way these TUN-B, so it's called a TUN-B dependent transporter because the way it works is that you have this protein TUN-B anchored in the inner membrane that reaches across the periplasmic space to grab on to this transporter and effectively cause it to open, allowing the substrate to come inside. And so the work that I did with Ahmad on this was try to understand this again using SMD. We had a new crystal, or he had obtained a new crystal structure of TUN-B, part of TUN-B, bound to the transporter. And so we used SMD pulling on TUN-B to see how it and how the transporter responded, how B2B responded. And so we found that there was a very strong connection between the two that allowed the transduction of force coming from the inner membrane ultimately to the outer membrane. And so this also resulted in a very nice publication, actually a couple in the end. But I say this actually predates the ribosome translocon work, so maybe we're just a little more exuberant, as you can see in this picture now, we're smiling. All right, so now you've already gotten a glimpse of the fact that gram-negative bacteria have not just one, but two membranes surrounding them. And so that brings me back to the question of membrane protein insertion. So we already talked about Sec-Y, so this is the protein responsible for insertion of membrane proteins to the inner membrane, but in fact there's a completely different complex responsible for them in the outer membrane. And so this complex is called the BAM complex, and in fact it also, one needs to acknowledge that another unique aspect is that while inner membrane proteins are exclusively alpha helical, almost all outer membrane proteins are beta pairs. So this also points to the need for a completely different mechanism of insertion. And so it was that initial work with Ahmad, looking at these outer membrane transporters that got me thinking more and more about the unique environment presented there. And so this is how we've started thinking about it in my lab. So zooming in on the cell envelope of E. coli, you have the inner membrane, you have the outer membrane. It has these crazy things called lipopolysaccharides with these O, in many cases, these O antigen chains that extend far above. You also have a cell wall in there anchored to the outer membrane in E. coli, and then a lot of periplasmic proteins. And so I just want to highlight maybe a few of the things we're doing with these models. So thinking about these outer membrane and periplasmic complexes. So for example, we're looking at multi-drug efflux, which is how it's one of the innate resistance mechanisms in bacteria, in gram-negative bacteria, where they can pump drugs all the way from the inside back out. We're also looking, for example, at how antibodies can bind to proteins in this outer membrane, navigating their way through this O antigen forest on the outside. And then what I'm going to spend probably the rest of my time talking about is the BAM complex, which is responsible for inserting outer membrane proteins. So this was a very schematic view of protein, of outer membrane protein folding around the time of 2011. So they knew most of the players involved. You have BAM-A, B, C, D, E in most organisms. And that this outer membrane protein, or UMP, must first cross the inner membrane. So it has to breach, it has to go through Sec-Y to get to the periplasm. at which point then there are some chaperones that bring it to this BAM complex where, we don't know the timing of all the steps, but somehow BAM helps it form this, this final beta barrel state in the membrane. So up until a few years ago, what was known structurally about these complexes, many of the players, BAM-B, C, D, E, these are all lipoproteins, so they're anchored to the outer membrane, inner leaflet. And then they knew part of the structure of BAM-A, that it had these paraplasmic domains called Pocher domains. But the key piece, BAM-A, that was the last one that was solved. And so in 2013, well, actually, the structures were solved in 2012 in Susan Buchanan's lab at NIH. So her and her postdoc, Nick Noynash, solved the structures of 2 species of BAM-A. So this one is full length. This one, in the structure, they were missing the first 3 Pocher domains, but it also would normally have 5. And what would, from first looking at the structures, it wasn't apparent, how this might be, aiding insertion. I mean, at first glance, it looks just like a beta barrel. Maybe it doesn't do anything. Maybe it's just another fancy way of anchoring these other domains. We don't know. Or at the time, we didn't know. But again, this was a collaboration actually started through a mod with Susan, but I was very fortunate that he let me continue it afterwards with her independently. And so I took this structure of BAM-A, actually both structures that they had solved, and simulated them. Okay, so the time, I had some Anton time, and so I ran it on Anton, and one of the things, so this is in a membrane with water, I'm just not showing, but we saw that this protein was extremely dynamic, particularly at this NC terminus, so this beta strand 1 and beta strand 16 would be opening and closing quite frequently in this microsecond time scale. And we also saw an exit port at the top open up periodically as well. So this was, we were able to reproduce this in simulations of both crystal structures under different conditions.
Is it the membrane or?
Yes, it's all in membrane. Actually, yeah, I'll highlight that. So I should emphasize it was in just a standard phospholipid bilayer. So DMP, I think I used at the time, and that was what we were using for our outer membrane mimic. So that's why I mentioned that it's somewhat permissive conditions. So sometimes we raise the temperature to 340, although that wasn't always required. Sometimes it would open up at 310. But it was always in this symmetric, simplified membrane model. Now, nonetheless, as Klaus often taught us, we should always seek out experimental collaboration and experimental confirmation of our results. And so, I was a bit on the edge of my seat when they did the experiments on this, but they were, Nick and Susan were able to confirm that this lateral opening was apparently functionally relevant. So when they induced cross-links between the 2 strands, the N and C termini strands, it killed the bacteria. So they were no longer viable. When they reduced that cross-link, then the bacteria did fine. So it wasn't a processing defect or anything. The BAM-A was getting to the membrane. It was just when you locked that gate closed, for whatever reason, it no longer could survive. Now, yeah, so Jerome brought up a good point. So that was all in just a standard phospholipid membrane, and as I briefly alluded to earlier, we know that outer membranes are actually quite different. And so we have moved now to doing most of our simulations in proper outer membrane models, where you have an inner leaflet composed of phospholipids and an outer leaflet composed of lipopolysaccharides. These, they're 5 to 6 aliphatic tails, lots of sugars, and in particular lots of phosphates. So they're highly charged, very large. They bind a lot of divalent ions inside this meshwork, and ultimately they don't move very much because of this. Nonetheless, though, so we continue doing these simulations now in these outer membrane models, and at least for one of the systems, admittedly at an elevated temperature, but to see this on a microsecond time scale, we were able to see spontaneous opening in this stiffer outer membrane. Now, that was for BAM-A from Neisseria gonorrhea. Now, admittedly, for the E. coli BAM-A, we didn't see it in 4 microseconds. But I'll come back to that. Okay. So another interesting feature that we observed in these simulations was a thinning of the membrane near this putative lateral gate of BAM-A. And in fact, in some cases, it was quite dramatic. I actually thought this was maybe a bit too extreme even, but my collaborators, Nick and Susan, really wanted to highlight that in the paper. But nonetheless, we always saw some thinning. And so I'm comparing it now to a homologous protein that doesn't have the same function, and there was no thinning between the two sides of the barrel. So that may point to another relevant, functionally relevant feature. And in fact, just reiterating, so we've repeated many of these in the outer membrane, and again, we see a pretty distinct thinning in the region near the lateral gate compared to the other side of the barrel. And this has been, we've been able to reproduce this in a number of different membranes, POPE, outer membrane model, even very thin membranes like DLPC. Occasionally we do see a little bit of thinning on the other side, but I think what's critical is that always near the gate, there is some thinning. Now, of course, as crystallographers and many others know, once you solve one structure, it makes all the subsequent ones of that same protein easier to solve. And so after Nick and Susan released their structure in 2013, a few others came shortly after. So here are the two from Nick and Susan, and I was somewhere in the middle of that. And then 2 more from E. coli came in the year after that. Okay, so more or less, at least at first glance, they all look the same. Certainly, we have different number of poacher domains depending on either the construct they made or whether it was resolved or not. But the barrel, at least, is pretty similar in all cases, except for one feature I want to draw your attention to. One of these, this C-terminal strand adopts different conformations in different structures. So in 2 of them, it kinks inward, and in 2 of them, it's fully zipped. So we call these the fully zipped ones, where it forms hydrogen bonds along the full length, and we call these 2 the kinked ones, where it bends into the barrel. And there's actually a reason for that. There's often a charged, or in some cases, hydrophilic residue at the end of this that normally would be pointing out towards the membrane, and so it's not very favorable in that conformation. We think that's why it wants to kink inward. So I mentioned I would come back to the question about opening, lateral gate opening, and so equilibrium simulations are nice for giving us ideas, but particularly given their length, they're not always nice for really quantifying rare events. So we turned to free energy calculations more recently in my lab. And so I'm comparing here now BAM-A with this kinked C-terminal strand versus one with it fully zipped along its length. And then comparing to a homologous protein that I mentioned before, FHAC, that doesn't have the same function. And so we just did umbrella sampling calculations, forcing open the gate and calculating the energy required for that process. And so what we saw was that for the kinked BAM-A, it's pretty easy to open. So I think, I don't have the number here, but I think this state would probably be, somewhere around this range, 10 to 12 angstroms maybe. So not too bad. If it's zipped, it's significantly harder to open, so we're getting about 10 to 15 kcal per mole higher to open it up. Makes sense. I mean, there's more hydrogen bonds you have to break. And then for the homologous protein, it's even harder to open. So it seems this one even goes beyond just having a certain number of hydrogen bonds you have to break, but there's something intrinsic to BAM-A that makes it a little bit more pliable. Now, okay, so I'm highlighting the importance of this kinking, but how can we be convinced that the kinked one is the native versus the zipped one? So certainly in crystal structures, we have two of each, or in the available crystal structures. So we decided so for there we did look at equilibrium simulations again. We went back and revisited some of them that we had run, and we saw that even the ones that started fully zipped eventually kinked. So within a few hundred nanoseconds, for example, this E. coli one here, so this is the zipped state, 180 degrees along the length. This is the kinked state down here. And so within, say, 200 to 300 nanoseconds, we saw it kink spontaneously. Same thing for the other one that started zipped, and they never come back. So they always remain in this kink state. So, it's not definitive by any means, but it suggests that the kinking is the more natural state and is perhaps functionally relevant based on our free energy calculations. So of course, though, we went to our experimental collaborators. Now, in this case, Nick Neunaj had started his own lab at Purdue. Susan Buchanan very kindly let him take the project with him. And so we turned to him and said, well, could you investigate this kinking for us in some way? And so what they came up with was to take a key glycine near the end of the strand, the hinge point of the strand, and mutate it to something bulky and see how does that affect the viability of the cells of the E. coli. So, so these are antibiotic challenge experiments. So it didn't kill them completely in this case, whereas the locking the gate closed had, but it did make them, in the case where you have a bulkier residue, it did make them more sensitive to antibiotics. And whereas if that residue was much smaller, like alanine, it wasn't too bad, not too much of an impact compared to the wild type. And so again, this really suggests to us that this kinking feature is important in the function of BAM-A. And in fact, we've now done, just to confirm, we've done a microsecond simulation of G807V, so moving to a valine, and it doesn't kink in the slightest. Okay. So the simulations and experiments are really going back and forth between each other and confirming the results of the other. Okay, so I'm just giving a really brief overview of what we're doing, so I'll just show where we're going now. So this really, I think this starts to encapsulate or maybe pursue one of the visions of Klaus's, was to see proteins not individually, but as a society. Okay, and so now we're fortunate enough to have enough structural data that we can start to see how all these BAM complex members work together. And so one of the things we've done in my lab is actually take crystallographic data from 3 different papers, none of them having the complete structure of the complex in total, but we've been able to combine them and generate a structure of the entire BAM A, B, C, D, E complex. And now we've run a number of simulations of this complex, and we're trying to understand how the different components work together. So in putting it all together, there's actually, and I don't want to take credit for the next couple ideas I'm going to show, but there are a couple theories that have been out there over the last few years about how the BAM complex works. So one of them is that it's somehow that the membrane distortion induced by BAM-A is sufficient to accelerate insertion, so it's known experimentally that outer membrane proteins, at least small ones, can insert spontaneously, but they do it very slowly. So maybe all BAM has to do is perturb the membrane, and that's enough to make it to overcome the kinetic barrier preventing outer membrane proteins from getting folded. So we've kind of pushed more towards a more systematic model known as the hybrid barrel model, in which maybe the membrane deformation is playing a role, but perhaps more importantly, this open lateral gate can provide a template for inserting beta strands. And so, at least the way this works schematically is that you have the new beta barrel budding off from BAM A. So BAM A then returns to its closed state, and you have your new outer membrane protein. One of the nice things about this model is that it always maintains the difference between inside and outside of the barrel. So between the hydrophilic residues inside and the hydrophobic ones outside. Now, honestly, in my lab, we've moved a bit away from this. It's a very cute model, but it's probably a little bit too organized for biology, where you have to have all these beta strands come in one by one and just perfectly form up. Particularly if you imagine just the great variety of proteins that BAM A has to help insert. So we're kind of moving towards maybe a little bit more comprehensive model, where this is what my student Carl came up with. We call it the loop guiding model, where you still have some initial folding of the beta barrel in the periplasm. You still have the membrane defect that makes it easier to insert at this point. But then you also have the open gate that allows these highly charged loops an easier path through the membrane to get to the other side. So with that, I think I'll close. Honestly, I cannot possibly acknowledge everybody involved. I basically stopped when I ran out of space to put pictures. But of course, Ahmad, Chris, Benoit here. Collaborators on BAM, like Nick and Susan, on SEC, like Tom and Chris Akey, people in my lab at Georgia Tech. I'm always very proud to show this, that I have my lab and students working on these projects. And of course, I wouldn't dare try to shoehorn Klaus into this slide. So maybe I end with a very happy memory of my own. Oftentimes the group would take trips to go canoeing at Turkey Run in Indiana. And so this was a time in 2008. We all went there together. I'm here, Klaus is here, and many of the people here are also in this picture. And so I have to thank him for, I was just a kid from a small town in Illinois. I was already very lucky to get to come to the University of Illinois. And honestly, I had no idea what I was getting into, but he let me be a part of something really amazing, and I have to always thank him for that. And thank you.
We have time for a few questions.
So it's very hard. You have been looking at this protein for a long time. You have a huge asymmetry in the structure. On one side you have long loops, and then on the other side you have short, really short.
Right, the periplasmic loops are very short.
And not really
Yeah, so the extracellular loops are very long and the periplasmic loops are I see.
This is a common theme for chlorine,
Not necessarily. Actually, Joanna Slusky at Kansas has done a lot of work on the bioinformatics of this. One of the reasons that you see that, at least in some outer membrane proteins, is that they form trimers. So the short region is where they come together, and so that the long loops are on the outer edge. But obviously, in this case, it's apparently working alone, but maybe because of the proteins it's inserting, that's important, that it doesn't interact with them too much.
Not that you have the LPS,
Yeah. So I didn't show this, but basically I proved all our early work on D2B wrong, and then revised it. But the interactions between the LPS and the loops are very important for how it arranges them and their stability and conformation. Yeah. Thanks.
All right. So let's make this again. Speaker is Rommie Amaro. So Rommie is a graduate from the University of Illinois. This is going to be English. She received numerous awards. I will be, spend 5 minutes talking about it. But instead, that's Rommie. Rommie, we're looking forward to you.
Thanks, Alek. I'll go ahead and hook up just because I tend to move around. So, yeah, I'm happy to be here. I can't really say that it's necessarily a pleasure, but thank you for including me. And I'm looking forward to giving a bit of a different talk today than I normally give. I feel like I'm on some kind of crazy talk circuit lately. I guess this is part of the life of being a professor. But in any case, today I'm going to deviate fairly significantly off script just to in honor of Klaus and how he has impacted sort of my career. And so, but ultimately, everything I tell you about is going to be mostly around the sort of the theme of molecular modeling at the mesoscale. And you can see sort of Klaus kind of emerging from this. I feel like I see him in so much of my work. So actually, I was not formally advised by Klaus. We never published together. I actually, of course, as many of you here know, I was a graduate student with Schulten, and I was actually, we started working together when I was an undergraduate, working essentially on protein structure prediction. And I've actually photoshopped my face until Elijah. Because there are no photos of our group. Because I guess it predated iPhones, and I admittedly was bad with organization of photos. In any case, but so I was working with Dan, and the second paper I had, but one where, so I felt like a little bit of an outlier in Dan's group, I think, because I was doing work, as this paper showed, that was really very similar to what was going on in the resource. And my second paper here, which was actually joint with Amaro, and was really something that was only possible at the time through the collaboration with TCVG, was we were studying how ammonia essentially gets transported. It's actually this super fascinating protein that we had studied for some years. Ammonia is basically created in a reaction in this subunit here called His-H, and then it actually diffuses within the protein to participate in a subsequent reaction acting down here at the second site. And so we were actually following on the beautiful aquaporin work. We had sort of utilized similar mathematical and conceptual strategies to do these repeated pulling experiments using the computational equivalent of AFM, of course, here molecular dynamics, and using Jarzynski's identity with the second cumulative expansion to determine this free energy profile. And it was really cool work. And really turned me on to the power of these sort of of molecular dynamics in general. And then right around the time that paper was published, TCVG held its first summer school. And this is the picture. Gosh, it was so ambitious of an undertaking, how many students were there, and how the resource worked to really put on what was an incredible show. And somehow I was included in parts of this, and actually much of it was around the topic of small molecule parameterization. So Zan, I tag teamed with Zan in a couple of lectures, and here's an action shot, which I can't believe is actually up on YouTube, this whole lecture, on the same stage, different outfit, but there's Klaus and Zan, and they had been gracious enough to let me give a little mini lecture on some of the stuff that we had been working on. But that really, I don't know how to describe it, but it was like I always felt like I was part of somehow Klaus's lab and the family, and I always felt very welcome. And then we started to go on these traveling road shows. And so we were in 2004. I was so excited to be invited to go to Perth, Australia to give one of these workshops, and this was at a time when we were piling all of these Mac laptops into these giant suitcases, and this is Elizabeth, I think, and Fatima. Yeah, you can recognize Elizabeth there. And it was a lot of fun, but this is also, I don't know, Elizabeth, if you remember, we had to work very hard to replicate, all of the systems and make sure that these laptops, were going to perform to spec, of course, because nothing could go wrong when you were in the classroom with the students and running these tutorials. And we took a picture of this clock because it's now 3:35 in the morning before the lectures were starting, and we were just, we just have to make this work.
After a 36-hour flight.
Yeah. It was crunch time, but it came off pretty much without a hitch. You can see most folks were happy. This is another just terrific picture of us in Perth. Of course, Klaus and Zan and all of the students there, and then Elizabeth and I. Fatima must be in there somewhere, or not, but she was there. Oh, there she is. Yes, thank you. Good eye, Amaro. And then in 2005, we went to San Francisco. So this was just really, anyway, it was just so much. We had so much fun. This is just a fantastic picture. We rented bikes for the day. So of course, it was a lot of hard work. Mostly it was, I would say, hard work. But we also we took time to have fun. We biked all over San Francisco. That was terrific. And then, of course, here's just a group shot. And this shot, which I particularly love, because you can see how his arm is sort of stretched all the way around. Which is really sort of a photographic image of how he made me feel, which was just absolutely, without question, included. And so, okay, now I'm going to try to switch some science. So what I've sort of done is actually kind of enmeshed sort of the stories about Klaus with some science, of course. I'm not going to rip you completely off the science. But so many of you will recognize this type of plot where, and I think this was sort of something that, of course, was really part of the fabric of who Klaus was the excitement of the convergence, really, of high-performance computing with data and biological or chemical applications to do something really exciting and make a discovery. And I show this. Gosh, for years I've shown plots like this, and these systems here that I show really were Klaus's. And then this one here was one that we worked on together over the course of many years, which I'll tell you about. But so. Of course, everyone in here knows about the analogy to the computational microscope, but I think this was really a beautiful way to describe these methods, and it's something that has resonated with so many people and is a very effective way to do it. So when much of my graduate work was essentially using the computational microscope in various ways to look at allosteric communication and conduction of these substrates to proteins. And then when I went off as for a postdoc, I decided I wanted to do something slightly more applied, and went to postdoc with Amy McCammon, and really focused the early part of my independent work on trying to harness essentially the sort of structural ensembles coming from these molecular dynamic simulations in drug discovery programs. And one very nice example that we worked on for years, which I'll tell you about just briefly because I think it illustrates the point quite nicely, has to do with p53 reactivation. So p53 is one of these central tumor suppressor proteins in cells. It's also called the guardian of the genome. In functioning cells, it senses cell damage and upregulates pathways related to programmed cell death, essentially, and cell senescence. And so a long-standing dream of, and what happens in over 50% of human cancers is that p53 is mutated, it has one single point mutation, and then is inactive. And so it doesn't, essentially these cells then can proliferate, they can proliferate, metastasize, and so forth. So a long-standing dream of cancer biologists has been to develop small molecule drugs that would bind to and reactivate p53, and therefore abrogate or regress tumor growth. And so this has been shown in various models, and is considered, even though it's at this point considered, I think, an undruggable target, it's something that still has a lot of promise. And there were a number of compounds after some 20 years of research on this target. There was a handful of small molecule compounds that were found to reactivate p53. Many of these actually were covalently binding molecules that would covalently attach to the core domain, and they knew this was happening, but they couldn't figure out why. So, one of the reasons, that molecular dynamic simulations has been so powerful is because it has the potential to, as we all know here, to elucidate sort of neighboring, at least low-lying substates of these drug targets that in some cases actually provide alternate routes for small molecule binding. And so here what I'm showing is a molecular dynamic simulation output of the P53 core domain, and I'm drawing your attention to one particular cysteine, it's the cysteine 124A, and all I want to really sort of indicate here is that it's quite mobile around this site, and you can see the formation, the transient formation of a pocket. And so what happens, what we saw, if you look at the crystal structures, and there's at this point now over 100 of them in the protein data bank, every single one of those around the cysteine 124A for the wild type P53 has basically the cysteine sort of tucked back, occluded from solvent, no real pocket available for binding. And in the molecular dynamic simulation, what we saw was basically this new site in that area opening up. And we used other algorithms to show, so the site opens, and then we can use other algorithms like computational solvent mapping, which I won't go into today because I don't have that much time. But this basically sort of allows us to assess the druggability of these sites or their propensity to bind ligand functional groups. So we find a pocket, we get hints that it's druggable, and then we sort of target it in structure-based drug design campaigns, for example, using approaches like virtual screening. We did this for P53, where we took various structures from the molecular dynamics simulations and that we had access to experimentally. We had a small library of ligands at the time. We docked them using a program called Autodoctina, scored them, and experimentally tested about 24 compounds, and we found one compound here, acetic acid, that showed dose-dependent rescue of mammalian cancer cells, which was cool. So we found a new compound through this novel structural information that we got only through molecular dynamics. And then we also showed that this cysteine 124A actually was the primary point of attachment for one of those rescue compounds that has been in clinical trials. So we were able to, using molecular dynamic simulations in drug discovery, we were able to not only discover a novel reactivation compound, but also rationalize the mechanism of action for a compound that was in clinical trials. So it's pretty cool. And then we significantly expanded our attempts to develop novel chemical matter targeting that site. And this actually formed the basis for a biotech startup called Actavalon, which is at JLabs over in La Jolla. And I am required to disclose that I am a co-founder of, have equity interest in, and I'm on the scientific advisory board of that company. So that's something that, I showed this here as an example for P53, but this is sort of now, I think, something that is an approach that is actually relatively, I won't say widely used, but pretty, it's pretty accepted as a way to explore these novel pockets. And then we also had developed, using an award from NVIDIA, sort of touching on GPU computing, sort of approaches using workflows to help others also do these types of experiments. Okay. Then in 2013, so around the time that was getting published, in 2013, there became open an opportunity to take over a P41 resource at the University of California, San Diego, called the National Biomedical Computation Resource. And this was actually a long-standing resource. I think it was not quite as old as Klaus's. I think it was 5 years younger than Klaus's. And it involved a whole bunch of terrific investigators. But the idea was they're doing multi-scale modeling and trying to develop technologies that allow us to cross these critical, capability gaps, as we call them, trying to bridge approaches, simulation approaches that will allow us to sort of really simulate and understand all the way from the atomic level to the molecular level, all the way through to whole organ scale modeling. And I somehow got in the mix of being possibly the person to direct it. And I was really excited about the possibility, possibly because I had seen all the excitement going on and been somewhat involved of what was going on at TCBG, but I wasn't totally on the inside, so I think I didn't see all the horrors. And now really familiar with. But in any case, so I have to say during this period that Klaus was very supportive and very helpful, and we had emails and phone calls, and he provided a letter that went into, so at this time when I took over was also when we were putting in the renewal. So the big thing about that was it allowed me to basically sort of create a vision that was my own, but of course then you also have to do the renewal. So it was a big pain in the butt. Anyway, so he provided this wonderful letter. And actually this is a slide from the site visit in 2013 that I used. And I showed this picture of Klaus and of me and the student. This was a picture from Perth. And one of the things that I discussed in the overview during the site visit was that, I had sort of felt like I grew up in one of these resources and that it was something that I really wanted to do and so forth. And so he was there. And then, oh my goodness. Oh yeah, so then we were like, well, what's going to happen? And it actually took quite a while before we found out that we were going to get funded. But one of the things that I learned through this process, that the reason why I put this in here, was because, and Klaus would follow up, how did it go, et cetera, and I said here, and this was really true, I have never been as exhausted as the day after the site visit, worse than running a marathon, which I'd run like 2 of, worse than having babies, of which I have 3. I have never been so tired. And he says here, and I just wanted to communicate that because I was surprised to feel this way afterwards. And he says here, I know what exhaustion you mean. I turned after all site visits into a zombie and basically slept for a month inside that zombie. I never had the strength to organize a thank you party for everybody. The party always came months too late, but they forgave me. And, it was just very nice. And then because we both had these these centers, then we also had the opportunity to work together in this capacity as resource directors, which was super cool. And in 2015, then we were able to have a joint workshop together on the topic, and Christian Poe was there. Also, there were a bunch of folks there on the topic generally of simulation-based drug discovery. And here's a picture of the Fahdis here. There's a picture of the group. He was just always so supportive and excited. It would be so cool to have our 2 centers work together. It's like, yeah, this is going to be fun. We did have fun. Oh my goodness. And then I would say also in terms of the technical parts of it, we wanted to get, for example, milestoning. We were interested, as he was also in getting milestoning into NAMD, because we were developing this hybrid MD-BD simulation tool. And again, here he's just absolutely nothing but supportive. And this work, which presents the computational framework for this new multi-scale simulation methodology, again, combining molecular dynamic simulations with Brownian dynamic simulations using milestoning theory, was published earlier this year, and actually is in his memorial issue in JPCP. Okay, so so at MBCR, one of the things that we've been very excited about, or that I personally also have been very excited about, because we actually work on a lot of different things at the center, but my particular real deep area of interest was sort of in extending what we know and can learn about using these sort of MD-based models or methods for single drug targets. But how about if can we really try to understand the drug target in a more realistic environment so that we can ultimately understand the molecular and chemical mechanisms underlying disease? And so I guess the rest of my talk is going to basically discuss our efforts to really bridge molecular modeling into subcellular scales and beyond, and the challenges there. So, of course, the technical challenges are, for example, multimodal data integration to build structurally realistic and detailed models. So, for example, combining, I took out all my nice sort of lead-up slides because I was thinking, I don't know how, I have still another 20 slides to go. So developing really structurally integrative models that combine, for example, very high resolution X-ray crystallography, possibly with cryo-electron microscopy data, tomography, et cetera, et cetera. And then also using information, for example, from proteomics and whatnot to actually develop more realistic in silico 3D models. And then once you build those models, however, they're going to be big, and we are going to be faced with the challenge of timescales. And so we need to be innovating in the space of new method development. And then at the same time, something that we all still have to deal with is, just as a field, how we can, our experiments become more and more complex. And that's difficult to keep track of and to really ensure rigor in, and that relates also to reproducibility. So at the center, we've been working actually for a number of years on developing a software called CellPath. And this is, I think, a pretty neat program that allows us to essentially construct these highly detailed three-dimensional representations of biological systems, and to do it in a way that is consistent with various types of observed data and/or hypotheses. And so, for example, you can take imaging data from tomography and use that to define surface containers, various kinds of surface containers. So this is actually HIV. And then if you know the sort of high resolution molecular ingredients and their stoichiometries, CellPath can construct for you a packed molecular model essentially at the mesoscale. So this has been developed by Graham Johnson, Mark Olson, Michelle Sanner over a number of years, always supported by the center, and we're working to extend it. The vision is, so you've seen these pictures, many pictures like this from David Goodsell of these beautiful, complex cellular scenes. The idea is to take a program that was originally essentially developed in the spirit of being able to create accurate graphical representations of biological space and actually connect it to different types of simulation programs. And so for just sort of summing this up again on the CellPath, because I think this is one of the more exciting parts of the problem that we're working on in terms of trying to bridge from the molecular to the mesoscale, is you can take all different types of input data, as I mentioned, high-resolution structural data, tomography data, fluorescence data, proteomics, etc., and you use all that data to construct your system restraints, so spatially in terms of defining compartments, and then you can also, for example, define different interior ingredients, their stoichiometries, where they should be constrained to be located, etc. This is sort of defining the interior ingredients in green and surface ingredients in red, and then CellPath can create models that are constrained to agree with whatever these parameters that you've input into. It can pretty rapidly actually create not just a single molecule or model, but actually a set of models or an ensemble of models. So it's something we continue to work on. So why it's useful for us, one example is with the influenza virus. And so for many years, us and others, including some of the highlights actually shown earlier today from Klaus's work, because he also worked a little bit on the flu. Many people had worked on just single glycoprotein studies using molecular dynamic simulations, and we basically wanted to move that to looking at whole virus representations. And so in collaboration with a lot of folks, but including Alistair Stephen for the structural data, he's a terrific collaborator at the NIH, we were able to reconstruct, this is sort of going through that, the influenza virus at the atomic level, using, directly deriving from the experimental data. And so one of the things that, of course, I'm sure many of you remember Klaus talking about, but that we're always faced with when you build these big systems is people really don't like big systems generally. I feel like I can pretty confidently say that now, and I think he would have agreed. There's always you're going to build this big system, it's going to take you so long to build it, and then it's going to wiggle for like how long, and what are you going to see? And this was always this is one of the central challenges, I think, that we sort of have to really wrangle. And so here I'm sort of elaborating, so I worked so hard to say, okay, why do we need to do this? So it does give you an improved sense of the physical arrangement of the biological entities in their complex environment, and that's often helpful. Just even a static representation of knowing how close things are, how they pack. It enables, if you are actually going to simulate it, actually enables simultaneous study of multiple components, because you have all these different components already embedded in the system. You can then essentially deconstruct those components and learn about, however many proteins you have in one simulation. The other thing is that you don't necessarily have to do all atom MD for these large systems. You can, once you go through the pain of creating these accurate, detailed models, you can actually use them as a platform for other simulation approaches like Brownian dynamics, Monte Carlo type approaches, etc. And what we sort of all know and have long argued, I think many of us are in similar spirit in this room, and particularly Klaus, was that really all the above will lead us to new avenues of investigation that just aren't possible on a single protein scale. That things are revealed when you look at their whole, that you can learn for perhaps about emergent behavior and so forth, that you simply cannot if you just look at the single system. And so, I had the opportunity to come in April of 2016 to the resource and give a talk on the work. And I remember I gave this talk, I talked about so much stuff that we do. And Klaus was there, which was great, and he was sitting in the back, and he had a scarf on. And he just said, he's like, Rommie, but why are you going to build this? And why are you going to simulate these ever larger systems? Essentially was what he asked. And I went through that big list that I had just put up. I said, it's going to tell us this, it's going to tell us that, it's going to tell us this, we're going to learn that. And he just said, no. I don't know if any of you guys remember it. I said, no, you do it because we can. And I was like, okay. Like, yes, I agree with you, right? But very difficult for that to pass a peer review panel. And I have to say that in the last couple of years, between 2014 and and now, I think that I've really come to an even greater appreciation of some of the battles that Klaus faced when he was trying to build and sort of realize his vision. But in any case, so in order to build these systems, we basically had to create new tools that allow us to construct them faithfully. And so we developed something called Lipid Wrapper, which basically allows us to develop all-atom lipid bilayers of variable compositions and then wrap them around any kind of geometry that we put in, which is great because then we can actually, actually instead of just simulating things with like a planar slab, we can have these nice membranes. For example, that's what we use to wrap the influenza virus, which looks spherical, but it's not spherical. It has variable shapes. And so, so this quest, so we were basically on a quest to simulate a full influenza virus. It had ultimately, after we built it, about 160 million atoms. I would say since the moment it occurred, since the moment we decided we wanted to do that, which was essentially in 2013, we were in touch with Klaus and the resource. It took us about a year to really to get the lipid wrapping tool going and the protein insertion tools and everything. It took us a year essentially to build the tool, and then you have to go through all the mechanics of how you apply for the computing grants. And here I have to again say that Klaus and his team were incredibly supportive. Many of you here in this room, it would not have been possible without your awesome help. And then so there's like a year or almost a year and a half we just were trying to go through Insight, through Blue Waters, and of course we were getting back these reviews like, oh, this is going to be great when exascale gets here, you're going to learn so much, but not at petascale, not right now, it's only going to wiggle and jiggle for like a few nanoseconds. Anyway, so I would say that there was really full support at that time. This is a picture from right around the time. We took 3 different trips back to UIUC to try to realize this dream. And I would say Klaus was extraordinarily generous. He let us use some of the discretionary time that he had been given on Blue Waters to do some benchmarking, and Jim was also extremely helpful, Jim Phillips. And I'd say we also got a lot of emotional support at that time. This is a quote from Klaus. He said, "You're Rommie." he said, I fully encourage you to work on this, but you need to know, small systems, small problems. Big systems, their problems multiply. They have big problems. We did, in fact, really find this to be the case with the membrane, but we got it to work in the end. In any case, they helped every step along the way. So we were able, ultimately, to run on blue water. So finally, we did actually get an allocation on blue waters. I think only thanks to a sympathetic program officer. So they gave us a little bit of time, and he said, Rommie, do something good with it. I said, okay. So we got it up and running. It had about 160, it has 160 or so, between 160 and 180 million atoms. We were able to get about 5 nanoseconds per day, and 160 nanoseconds of simulation was basically all that we could get. And this is the wiggling and jiggling of the virus that you see. You see all those little guys moving around, those little proteins. And so I'm spinning it, and we're looking at, I'm translating it, now we're going to go into it. But the wiggling and jiggling that you see, that is the trajectory from the MD simulation. I'm hiding all of the waters. The blue here, this is the membrane. Influenza is a fully enveloped, lipid-enveloped virus, which was something that Klaus was also very excited about. You can see all these different microproteins. Okay, so then it's like, well, what did you learn? So we first start looking at this, and we're thinking, okay, if we let's start with the basic analysis. First frame on top of the last frame, what do we see? Basically, so the first frame is this sort of solid color, and this glowing mesh is the last frame. And basically, you see that. Well, yeah, it doesn't really move that much. There's no large structural rearrangements. So I'm thinking, the reviewers are right, this is just a waste of time. No, but of course we weren't thinking of that. But in any case, we didn't actually expect that we were going to see any sort of large-scale structural rearrangements, and if we had, we would have thought something went drastically wrong. On top of that, influenza virus has a matrix underneath, so it's interacting with the tails of these microproteins, so I don't think much rearrangement is going to happen anyway. But I wish I could go back to April 2016, because I look back at those slides, and at the time there was something that we had not yet realized, which I think turned out to be very key for us. And so I would have liked to say, because we can, yes. And this is so important for study capabilities and, for so many reasons. But I wish that I could tell him today what some of the things that we know about these systems. And why do we do this? It turns out, because we've done extensive simulations of the individual glycoproteins, and now we have really great control comparisons to the whole virus, actually those glycoproteins, because they are packed in a fairly close environment in that virus, as you saw, they actually are interacting with their neighbors, and it does change. In fact, this is just looking at a principal component analysis of the neuraminidase proteins and the hemagglutinin glycoproteins, comparing the single glycoprotein principal component analysis with where we are going and sampling in the virus. And they do actually sample slightly different conformational space, which again isn't surprising, but it's something that people haven't really discussed. And the other thing, which was more, I think, of an intellectual contribution, is that what we can do now, because we have so many copies of the individual proteins inside these cell scale scenes, we can actually use Markov state model theory to discretize, essentially to discretize to deconstruct the virus. So we have hundreds of copies of the hemagglutinin. We have multiple dozens of copies of the neuraminidase. And instead of what people do for Markov state modeling is they're basically taking, in most cases for molecular nanosimulations, they're taking these single copy systems, spawning off tons of parallel ones of those, and then using this sort of network of states approach to define the metastable states, for example, and then you can sort of, I'm sure actually probably Benoit talked about this yesterday. But essentially what it allows us to do is to extract long timescale dynamics from many short timescale simulations. And so even though we only sampled for 150 nanoseconds of the virus, we can actually say things about the dynamics, the kinetics of these systems out to microsecond timescales. And so why do we care? Because, well, it turns out for the flu, this is characterizing the kinetics of protein motion and so forth, and ligand binding is like of interest, just broadly speaking, but in particular for the flu, do. There's a loop here right next to the drug site, and a lot of people have essentially made a lot of stink about this particular loop and the fact that as it moves, it can open up new opportunities for drug design. We now have characterized, using Markov state model theory, basically the mean first passage time for loop opening and closing for a couple of different loops on this particular target. And of course, we also get a lot of information about drug binding sites and so forth. So this sort of, I think, was a very nice way to, in some sense, get around this central criticism of, okay, well, you're going to have this big system, and what are you going to get out of it? This is one way that we can say, well, we can actually extract something truly meaningful from these systems. And I have to say, it has actually helped get subsequent proposals through the review process. So I think, in general, it's something that people are acknowledging. The other thing that I touched on earlier that I just wanted to mention again is that once we construct these systems, we don't just have to use molecular dynamic simulations. We can also use things like rigid body Brownian dynamics, which, for example, here allows us, like, for example, with the flu, we're interested in looking at how drugs diffuse and bind to different sites on these glycoproteins. And so this is sort of showing how we can really, it allows us to essentially cross scale. So the virus itself is about 100 to 120 nanometers in diameter. And you can see the drug, actually, what we can do is this is again using rigid body diffusion sort of Brownian dynamic simulations. These drugs can bind to any one of these sites sort of highlighted here. And we go all the way down. When we're doing these types of simulations, we are actually looking at the very intricate type of interactions at the atomic level when these molecules get close. And again, why we care about that, for example, with flu, we were able to reconstruct 4 different strains of the flu virus that replicate essentially strains that they have found in the wild, or clinically. So we can look at the swine flu, the bird flu, and then other lab-created strains. And these strains have different features, which people who influenza biologists have long wondered about. So, for example, looking down here at this bottom panel, this is a slice through the virus. You can see that the blue guys here that looks like a peanut, this is the hemagglutinin. The guy that looks like a broccoli, this white molecule here is neuraminidase. Again, here's the hemagglutinin, and here now this really looks like broccoli, is neuraminidase. But you can see that in some strains of the flu, in fact, the most virulent ones, like the bird flu, this molecule has what they call is a stop deletion, and you can see that it's actually much shorter. And it has a very different type of electrostatic profile when it's the virus has a very different electrostatic profile when it has these variations in the proteins. And so we were able to basically construct in silico these different strains and look at how patterning of these molecules as well as height actually affects the association rate of what we call sort of mimics, so sort of substrate mimics, essentially, sialic acid to these different sites. And one of the things that we found that was really unexpected is that, and I'm running out of time here, so I'll just wrap it up, but is that influenza folks have long wondered why there is a secondary, it's called the secondary sialic acid binding site on the neuraminidase glycoprotein. And we actually think that it has something to do with, well, actually, that it has this secondary site has the fastest rate of dissociation of these host cell substrates, which was an interesting finding. And also, there's, I'm trying to say too much in too little time, but we put forth a hypothesis about the relative rate of association to the different sites and linking that to transmissibility of the different strains. Anyway, so there's always a lot of folks to thank, in all of this work, but really, this symposium and most of this talk has really been inspired by Klaus and his lab and the resource and many folks. And I think, I think when I think about his legacy, I mean, it will have, so many components to it. But certainly the ability to build these atomic level systems or these atomic level systems, but at a tremendous scale, that will only increase to something that I think is really a strong part of his legacy. And so, even in the past 5 years since I was at the resource, we've really gone from, tens of nanometer scale to hundreds of nanometer scale for these types of systems, and now we're building systems that are on the order of microns at the atomic level. This has, getting ready for sort of the next big computing architecture, and this is just a dendritic spine. You can see the actin gel within that dendritic spine, and we actually do now have we're building these models of the next scale. So this is a kind of one of the scientific legacies that certainly will continue, and we'll be proudly carrying it forward. That's all I have.
We can have one or two shows.
Yeah, of course, a lot of great work. I was wondering one thing about P53 work, which is how many different commonly occurring mutations do you see in P53 across different cancers?
Yeah.
And how generally effective is any given reactivation?
Okay, I can talk about those details. So there is actually about there really are six, well, there are many mutations, but like 90% of the mutational, for the core domain, there's about 6 major ones. And so how effective are they?
Well, is there any one reactivating agent that works against multiple?
Okay, yeah. And so that's one of the strategies that they're trying to push forward at Exelon. But yeah, there are agents that will reactivate multiple mutants, and then there's also reagents that are mutant specific. And it's in part because actually the dynamical ensemble of each of those mutations, the mutations basically destabilize that core domain. And so they, in some sense, start to become a little bit unfolded, and they become essentially unfolded in different ways. So the sort of, the sense is that there might be druggable pockets in one particular mutant form that is unique to that mutant form, but that there are other pockets that actually where you can develop broadly reactivating agents for.
All right, well, let's thank Rommie. We have now our short video that's an interview with the Klaus Beckman Institute, and after that, let's see that
I'm Klaus Schulten. I'm a physicist who uses his concepts and methods to study living cells. I built over the years what I like to call a computational microscope made of concepts and ideas from physics and chemistry and mathematics, and made of software and computers that put those ideas and concepts together to simulate what is happening in living cells. The computer can actually follow, at the level of the atoms, the motion of the molecular machines in the cell. And so that is a tremendous advance over static pictures. You can basically see the action, and living systems are characterized by moving. There's a beauty in the living world, not only in the living world that we see when we go to the zoo or go into the countryside, but there's also a beauty further down at much higher resolution. When you see a cell, you are impressed with the liveliness of it. You see things streaming and moving, and bubbling and sculpting, and so that is life, and that's what we want to show the world. And so I'm just in love with having seen this myself for the first time, and I'm very proud that my excitement is shared by other people, but I'm doing my work because of a keen interest in how nature works. So with this kind of motivation, I'm of course very happy that I don't see myself isolated in this world, but that I see that my excitement is shared by people who want to know what is really there.
Well, I really feel deeply honored to be included in Klaus's family. Klaus really, for more than 2 decades now, I know him in person, and it's been very, very dear and close to me for all these many years. And in many capacities, and one is the obvious one is a scientific leader of our community, and I think it's fair to say, many often used, but he's a visionary in the most positive sense. He has pushed us in the field into new directions that have come eventually, but thanks to Klaus, they have come years earlier than they would have otherwise, because he provided not just the visions, but also the tools, the means to get there, and we've seen beautiful examples of this in this meeting. He's also very directly involved in guiding science, and in particular, I'm also here as a representative of our Max Planck Institute of Biophysics in Frankfurt, where Klaus is very much involved, and he has been on the scientific advisory board, playing a very active and positive role in steering us into the future. Klaus has also been always a mentor to me, and a guiding force in a very personal way, and most importantly, he's been a friend. Here's a dinner at Amaro. Recognize Attila, with somewhat longer hair, and this is Bill Ethan, and there is Klaus, and I have this picture of the people on the other side. I'm not going to show this today. So as I said, I met Klaus in 1997 at the house of Joel Berenson, and I was still at Los Alamos. We were at the time both working on cytochrome oxidase. Very one of these events, one of these dinners that keep the memories for a very, very long time. And so it's very special. But on a lighter note, we're supposed to be also somewhat entertaining here. That I saw also interesting sides of Klaus that some have already been touched on. Certainly, as an Austrian, I'm not easily impressed, in particular not by a German. And so when you guys talk about transputers, as it happens, also in Austria we did transputers, and I was programming in Occam before I was programming in Fortran. So, but it's science, big systems, Klaus. I was amazed, but as I said, not easily, but what always impressed me with Klaus was that his ability to go shopping. And you already heard about cashmere sweatpants. My second really close encounter with Klaus was at a meeting in Helsinki, I think 98, maybe 99 in the summer, and Klaus went shopping to Marimekko. I'm trying the best Finnish I can, and for those of you who are not fashion aficionados, this is about the most colorful combination of clothes and interiors you can imagine. Klaus was going in there, and I was wondering why in the world would he do that, and it turns out it was that. So the second thing that impressed me, as I said, it's not the science, I'm not impressible. The second thing that impressed me was also at this meeting in Helsinki. Klaus is an early adopter, and I think we have heard this in the review and NVIDIA talk yesterday. He's an early adopter of computers and everything. And contrast, I'm a late adopter. I get an iPhone when it's already, and my kids are already asking for a Samsung. So at this Helsinki meeting, Klaus was not only using PowerPoint, but when he walked up to the podium, it turns out he had taken a picture with his computer camera of the speaker slides before him and quickly integrated them into his talk, so to attack with more finesse. So that really impressed me. And with that, I over to the scientific side, and so I will try to address two questions today. One connected to certain interests that Klaus had, and I think is an interesting question, because I will show it's a physics question, and that is a biological physics question, and that is how do cells, living cells, know that their membranes are in good shape? And the second question, how do cells shape their membranes? And of course, shape now used in somewhat different ways. And what I will use, what the plan is to use Klaus's computational microscope. And I remember when also this was actually at this dinner, when I showed the picture from was Klaus coming to the NIH and giving, this was a computational microscope talk, a grand talk at the NIH, and he was super enthusiastic. And I think this was an uplifting moment for all of us. And so I will do something in a somewhat heretic manner. I will set the microscope to 3 levels of magnifications. And of course, for Klaus, there was only one level, which was all atom or full quantum. But okay, so membranes, I think in this community, long before I got interested in membranes here in Urbana, everybody was interested in membranes because smart people realized that in cells, lots of interesting action happens in membranes or around membranes. And again, beautiful presentations at the meeting before. Here's a cartoon rendering of the endoplasmic reticulum. It's already outdated. There are thanks to high-resolution optical and electron microscopy, much finer details. These are also tubular structures. And just to give you an idea of why cells obviously must be investing a lot of their energy into them, and we also know that if your cells have membrane stress, or if you have certain medical conditions, metabolic disease being one of them, your cells often do have membrane stress, and so membrane per se, and the plastic reticulum in particular, are associated with also disease conditions, neurodegenerative and metabolic diseases. And one of the stresses that I have gotten into thinking about, thanks to a collaboration with a colleague in Frankfurt, Robert Ernst, is when your membranes have too many saturated lipids, and membrane viscosity goes up and the mobility goes down. And one way of certainly microorganisms, but as I was just told by also medical researchers in the field, also in higher organisms, one way of pushing into this state is by eating pizza. We had butter-rich pizza for lunch, and so I think we are on this side. So, but yeast can easily fix that. Yeast has a pathway that gets activated of the desaturase that takes fatty acids and induces other ones. And so the question was, how is this pathway activated? And we know from lots of preceding studies what the molecule is that does this. This is one of the activators is a protein called MGA2. There's a second one which is, I think, very similar. And so MGA2, just to for the more physically minded people, and as I hinted at, I was also trained in physics, to give you an idea of how beautiful and confusing some of the biological solutions are. So what we know about MGA2 activation is that through a mechanism that I will try to describe, it gets ubiquitinated. And the ubiquitin chain, growing ubiquitin chain, recruits the proteasome that Wolfgang introduced to us earlier in the meeting. And the proteasome degrades part of the protein, the linker and the transmembrane leaf, but not the rest of the protein. The rest of the protein actually gets liberated to another cofactor, and the cytosol is taken up into the nucleus and acts as a transcription factor. So that's, I think, quite interesting. The question now is what activates this MGA2 molecule to become ubiquitinated and then trigger the downstream activation of the only one pathway. And what we know is this activation responds to a variety of changes, lipid composition, cholesterol, and so on. And for those thinking about biological membranes, these are not POPC membranes. They have 60 or more different lipids, cholesterol, and all kinds of additional cofactors, proteins. So if, an example I tried to give, if somebody gave you a very precise mass spec fingerprint of everything that is in your membrane, and you had to decide what is the phase and the state of the membrane, that would be a rather difficult challenge. You have to go a 60-component phase diagram with some of the components fractions that are really, really small, but still have an influence. It's impossible. The cell has the same problem, and that's why I think of this as a physics problem, because chemistry cannot help. You can, even if you did a complete chemical analysis, you could not know with precision where you are, because these phase boundaries are actually usually very close. So what is then the mechanism? And there have been ideas, and of course, the first one that comes to mind, I was already drawing MGA2 as a dimer, so maybe it's dimerization, but that could be ruled out by our collaborators because they just overexpress MGA2 extensively and has no effect. Still responds in the same way to changes in their case, the growth medium. However, a single amino acid mutations of a tryptophan has no effect on dimerization, but blocks activation of the pathway. So that suggests a different mechanism. So one possibility that comes from the bacterial world is the hydrophobic mismatch. So this is a process, a mechanism used in bacteria because their membranes have a well-defined thickness, and if you change that thickness, you can use scissoring. But that can also be ruled out because our collaborators, again, they introduced different numbers of leucine residues in the center of the membrane, and if they added one full turn, the activity was back to normal. And it turns out that there was a variation in the activity as you go around the phase angle. That also turned out to be important, so I mentioned it here. But this goes to Klaus. Experimentalists, when they have run out of options, they talk to computational people. And I don't say that, Robert Ernst has been interested in us, but doing this problem with us even before, but certainly that was the point that our collaboration got serious. And this is what triggered a number of simulations, and of course I'm not showing you the ones that did not work. I only show the ones that when we finally got headways into the problem. But the simulations show, these are simulations done by Roberto Covino, a postdoc with an even more physics background than mine, thrown into studying membrane systems, really learning as he went along. But he observed these are coarse-grained simulations, because missing simulations failed. What he observed was the two helices, that this is a single-span transmembrane protein, the two helices dimerized, that's good. And what he also observed was in our long simulation, there's something unusual that caught us a bit by surprise. That these helices, once they had dimerized, kept rotating around each other. Normally, you run simulations, if two proteins have found each other, they stick. There's not much going on, even if it's not a good binding site, but and if the protein has a good binding site, or the protein would always eventually will lock in. But here, they kept on rotating, although the rotation is very slow. It's a very long simulation. This run is 1.4 milliseconds, which is very long, even for a coarse-grained Martini model. And the lifetime of the different rotational states is about 10-ish microseconds. So you have to be patient. This is even on a sizable computer system, rather long calculation. What he then observed was, looked at the structures, there were dominant states. One had this tryptophan, I already mentioned that if mutated to alanine abolishes function, tryptophans pointing towards each other. The other one had the tryptophans pointing away from each other, and then there's two states that are symmetry equivalent, one in, one out, that are in between. And what was also a clue now as to the possible function was that the population of these two states in simulations with different lipid compositions appear to depend on the level of saturation. In a more saturated lipid environment, in the buttery orange, POPC, with a single double bond in two chains, we have a slight but significant increase in the population of this state. In the olive oily green lipid composition, I point this out because there will be a green and orange coming up later in the talk. Then this olive oily state with two double bonds, we have a preference for the other state with the two tryptophans pointing out. And what with that hint, our collaborators did now a number of physical characterizations, and one of them, I'm going to show here, this uses EPR, the parametric resonance spectroscopy, to look at the proximity of spin labels. So we place spin labels all along the peptides, and then look at their distance as a function of lipid composition. And the lipid composition is here ordered using a fluorescent dye that is sensitive to packing, C-Lorden, from left to right. So left is fluffy membranes, right is tightly packed membrane. And this you can just read off by the color of this dye. And the correlation between distance and order is essentially perfect. That was very suggestive. There was other measurements. What they suggested consistently, and also this leucine insertion, that we are dealing with a rotary sensor. So this molecule seems to, at least the helices seem to rotate against each other. So that's, could be just coincidence, but certainly very suggestive. So why would these rotations occur? Why would it be sensitive? So if you look at the structure, so we did also long all-atom simulations, lifting some of the coarse-grained structures, and you see the result of one of these long simulations with the on-stage of the fence pointing in. Now you can see there's another cluster of aromatic residues at the bottom here, and each of them are pointing in, and they're packed away. So suggestion by this picture is that we have a packing sensor where the tryptophan residues are very big. Point out in lobby that there's a big perturbation in a dense membrane, but they are relatively easily accommodated in a fluffy membrane with high content of unsaturated lipids. So here is the Swiss cheese model of this. If you have holes in the membrane, knobs can point out. If you have a thick bordered membrane, then with things are tightly packed against each other, then you want to present as smooth a surface to the outside as possible. So the prediction of this model was size matters. And size here would be the volume of the amino acids. So that's what our collaborators then tested. They varied the tryptophan, introduced a number of different side chains, and what you can see here is these are now live cell experiments. They actually looked at the activation of the OE1 pathway in the different mutants, and what you find is that the activation depends, again, in an almost perfectly correlated manner on the size of the side chain. That was so is reassuring that we're not just dealing with some biophysics type mechanism here, but we're actually looking at something that is related to the in vivo sensing function of these genes. So this is another result that I found quite stunning. This is going back to this mutation that I mentioned earlier, W1042 to alanine, so the tryptophan replaced by alanine. As I told you, if you do this in yeast, the sensor is completely dead, no activation. And it turns out that we can recapitulate this with an empty simulation model. So here you see the distance distribution between the alpha carbons of these sensory tryptophans in the buttery and the oily membrane. And you can see in the wild type, there's a big difference. And in the butter, the on state, the in state is favored, and in the olive oil, the off state, out is slightly favored. In the mutant, two things have happened. You're now always in the off state. There's almost no population in the on state, consistent with the prediction of the model. The sensor is off, not activated. And on top of that, the sensing ability is gone. The population green and orange are now identical. So this strongly suggests that this is indeed not just sensor, that it is the law, that it is the sensing mechanism. Still mysteries, the question of how you go from rotation to signal transmission, ubiquitination, and so on, but at least it's very molecular, and I think simulations have helped us understand what's going on. Second system that I want to talk about is another sensor, and this is considered more important. So MGA2 is in yeast, although I think that there are indications that similar processes might also be happening in higher organisms. But IRE1 definitely is a sensor that concerns us as well. It's associated with again, metabolic diseases, diabetes in particular. IRE1 is associated with unfolded protein response. It's been studied by Peter Walter and collaborators for many years, and really great detail with spectacular results. And one of the interesting findings that you can see in the literature, but it's not discussed in great detail yet, is that you can activate unfolded protein response by lipids. Now that sounds counterintuitive, because obviously lipid bilayer stress doesn't seem to have much to do with unfolded protein response, but there are strong indications that bilayer stress in the ER and unfolded protein response intersect, and they intersect in this protein. But before I explain to you what we think is happening at the molecular level, again, this is for physics-minded people. Another example of the other solutions that nature has come up with that seem to have nothing to do with the MgA2 solution. So how does IRE1 sense stress? So it senses stress by oligomerization. So if you have clusters of IRE1, then they activate each other. They activate each other because they have a kinase domain, and that leads to transport phosphorylation, so they phosphorylate each other. Phosphorylated the veins and the cytosolic site activate another domain at the bottom here, which is a ribonuclease domain, and which is inactive with the absence of phosphorylation. In the activated ribonuclease acts on a messenger RNA. It does an unconventional splicing of this particular messenger RNA. So it cuts out a particular piece of the messenger RNA when activated, and that now unconventional splicing of the messenger RNA, the gene product of that is the transcription factor. Again, that is taken up into the nucleus, activates this gene cluster associated with the unfolded protein response. I think that, to me, this is just one of the beauties and surprises of biology that in physics you just rule this out. So back to the theme, what drives oligomerization? So it's easy to explain why unfolded protein drives oligomerization, because there is a domain on in the lumen of the R that is combined unfolded peptide chains, and many of them there you have clusters of IRE1. But what is less understood, or not understood until we started this project, was how the activation could come by changing the lipid composition. Again, you have a single span transmembrane helix, but in this case, it has a very unusual structure. So previously we had this tryptophan at the center, and MgA2, the tryptophan at the center, which is also unusual, but here it's even more unusual. This helix has a strongly hydrophobic, typical tail on one side, and then it has a very clearly antipathic helix on the other side, with a number of charged residues on one face of the helix. Mutations here in this antipathic helix, so this valine mutated to arginine, affect both the unfolded protein response and the lipid-induced response. If you change again the growth medium to a medium-rich in inositol, you can also trigger unfolded protein response. The effect of the mutation, the magnitude of the effect is essentially identical whether you do a standard UPR stress using redox stress, or whether you need to incorrect crosslinks, cysteine crosslinks, or whether you change the lipid protein here. That suggests indeed this is intersecting, and the mechanism affects, or this mutation affects both processes. So now, same problem, how does this work? And again, simulation gave us at least a clue. So here's the wild-type helix placed in a membrane, and a number of simulations with different lipid compositions. I'll show some results, but I showed just one movie. And what you can see is this helix does something rather unusual. Instead of staying nice and straight, or maybe slightly tilted, it lies in this membrane almost flat, maybe with a slight kink here. And the reason, of course, is that this top part is antipathic, and there's this big hole here where the charge cluster pulls in a pocket of water molecules. This really perturbs the membrane structure around the helix in this top antipathic region quite dramatically. And by comparison, this mutation where into arginine, the helix stays straight in all membrane compositions. The arginine snorkels to the top, and the top part of the helix actually unfolds, which is something that our collaborators confirmed sees spectroscopy, that the helix content of the mutant is indeed reduced. So why do we now think this perturbation can be used for sensing? What we found was that the extent of the perturbation, both measured by variations in the thickness of the bilayer, how far out is the thickness of the bilayer perturbed, and by the order parameter of the tails probed by NMR spectroscopy, the extent of this perturbation, the spatial extent of the perturbation depends on the state of the membrane. In a thick, highly viscous, ordered, saturated bilayer, the extent is significantly larger than in a thinner, more fluid bilayer. This is the lines here of the 3.5% changes in there. So what we think is going on, but that's something that is currently under investigation to a little more detail, is that these helices act as defects in the membrane, and coalescing the defects lowers the overall energy. You have in some less perturbation, and and in relative terms, the cost of the defect is higher in a thick ordered membrane than in an already fluffy, easy to deform membrane, and therefore the driving force for aggregation or oligomerization is higher in the thick membrane. That's our working model, and in the mutant, this ability is lost because the straight helix is about equally happy in all conditions. And so we have one pretty good experiment to back this up, and this is again using EPR spectroscopy to look at spatial proximity using a variety of spin label positions, and this is now using helices with maltose binding protein spin labels in vesicles, and varying again the composition, probing them, the viscosity or the packing density, I should say, with a fluorescent light, and again we find the more densely packed the membrane is, the closer the two helices are pushed together, the closer they are brought into proximity, as you would expect based on our oligomerization model. So we think that it certainly gives us an idea as to what is going on. Good. So, how am I doing with time? Okay. I promised something about membrane shaping. I will just give one story, no fusion. I wanted to show a bit of fusion, but maybe over coffee tomorrow. So one problem that I've been quite interested in is how these intricate shapes of membranes come about. We have thought a lot about tubular yard. There are theories and explanations out there, but they're actually also actively revised by the people involved in the field. It seems to be a very still active area of research. But one thing that I've been particularly interested in is the question of autophagy. And autophagy, for those not familiar with it, is the term means self-eating. It's a process that can be triggered by a number of factors. One of them is starvation. When the cell runs out of food, it starts to break down compartments. It's also a way of housekeeping. It's used, for instance, to break down mitochondria and other large structures, in synaptic for breaking down including bacteria and so on. And so one of the questions in autophagy is how this double membrane structure, so these red lines indicate a cut through a double membrane structure, there's a cup-shaped double membrane structure that engulfs its target, how this is originating. So it grows around the target, closes up, and then infuses with the lysosome. The question that we're concerned with is what happens at the beginning and end. What we know is that there is initially is fusion in yeast, at least of three vesicles based on a number of very nice microscopy experiments that triggers this process. And we also know that there is this protein A2G1 that appears to play a role. So We know there's about 10 ADG1 complexes localized at the 504 assembly site. So the idea was that these ADG1 complexes recruit the vesicles and then they fuse, and voila, you get the cup shape. And that was when Rommie, a postdoc in my group, once he realized, he's a membrane biophysicist, once he realized, oh, 3 vesicles, fusion, he knew that if you fuse 3 vesicles, conserving the volume, conserving the area, this shape is the energetically preferred shape. So this should happen automatically. So this, or think of a Microscale is a deflated soccer ball. So, but there's a problem. And the problem is that there are boundaries in shape space. So this is like boundaries in phase space. If you take a membrane and you change the volume to area ratio, the transitions between different shapes can be discontinued. And there's particular boundaries from the tube to the disc, from the disc to the cup. These are shown here using excess area as an order parameter. I'm not going to discuss this in detail. And you can see local minima corresponding to tubular shape and the disc shape be separated by barriers. These barriers are actually quite large. They can be 30 to 50 kT high. And that causes problems here, and this is illustrated with this animation. Yes. So we simulate now an even higher level of coarse grain with a triangulated fluid dynamic membrane. What happens if you fuse 3 vesicles, instead of going to the energetically preferred cup shape, you go to a tube. And so the tube is a kinetic trap. So we confirm this by also just taking a spherical vesicle and slowly changing the area to volume ratio. So you grow in an area, you wind up with tubes. And this is even though the driving force is dramatic. There is an enormous energetic gain to convert a tube into this double deflated sphere shape. And this driving force, by hundreds of kT in this case, would prefer that state, but there seems to be the barrier is insurmountable. And we confirm this by molecular simulations, using a coarse grain model with 3 vesicles. The problem persists also with 5 vesicles. So here we seem to have run into a problem, but I already mentioned to you there is this interesting protein ADG1 that is present here, and this is 40 nanometers, so where is Angela? This is as big as your HIV capsid, so this is a big protein. Building is here, so we saw the middle helices. This is really a very sizable protein, and the idea has been, I mentioned this is a recruitment protein, but we thought maybe it plays a role also in this process. So the idea was, can this be a factor converting the vesicles into the cup shape? So we built look at the structure, and it turns out that it has extensive basic patches that might be membrane interacting. We built a coarse grain model, and then combined that with this hyper coarse grain membrane model and ran simulations. And now we have spontaneous transitions from the after vesicle fusion to the cup shape. So the barriers are all gone. So you see what it does is this S-shaped structure, double crescent, fits extremely nicely into the cup, and it induces negative curvature on one side of the paddle shape, and then in a almost barrierless transition, and then they can cross over. So you can see in this chapter, you can see why this is such a good molecule to drive this transition. So our collaborators, this is a small collaboration with Jim Early at UC Berkeley, and when they learned about these simulations, they looked for possible mutation sites outside of known protein interaction sites and with relatively innocuous looking mutations at the surface. And these were changing charges in 2 to 3 amino acids at different points. And what they found consistently in the standard topology assay is that these charge variation and mutations, so introducing negative charge where you within basic patches lead to either complete abolition of autophagy in yeast or about 50% reduction in the autophagic activity. So with that, I want to wrap up. I hope I could convince you that, but of course maybe here I don't have to convince anybody that simulations are a good thing, that we can learn something, that we can also discover something. So I think this is really in Klaus's spirit, that simulations are just looking what happens in a simulation with any preconceived ideas as to what might be going on. You can discover something as you go through an experiment, and then you can go back and make your standard hypothesis-driven research, tested, validated, make predictions. Both for sensing and also for autophagy. We think ATG1 plays an interesting, more complex role than just recruitment and in the assembly process. And with that, I want to thank the people who did the work. Roberto Provino, I already mentioned, really a wonderful collaboration with Robert Ernst with that Stanford W3 position in Saarland, Anja Rommie, the JDR, simulation and autophagy simulations in collaboration with the group of protein programming. With that, I want to end and thank also Klaus. As I said, he really pushed this field into new directions, and I think he kept us honest. All the people I know in the simulation community, he was always the one who was looking for working closely with the experiment rather than staying in the ivory tower of pure simulations and an alanine peptide model system. Go out, try to do biology, and I think that for and the younger people, the students here, this is just the beginning. So, thank you.
Time for one short question.
What happened if you try to re-engineer the receptor by putting a tryptophan on the opposite side of the helix?
Yeah.
And it would turn off when it needs to turn on, or?
Interesting question. So there is another aromatic residue on the other side, it's an element, if you may have seen it. So we think it's balanced. So, but that could be an interesting thing to swap them.
Or you could tune it to overreact, or...
Yeah. So if you look at it, has a bit of this propeller thing, so it could be that indeed that could be enough to have the opposite phenotype suggestion. Thank you.
All right, let's our next speaker is Klaus Schulten from Beckman Institute.
So it's a great honor for me to be here and pay homage to Klaus. I changed the title of my talk a little bit after talking with some of my colleagues and friends here. It will become apparent in a minute why I chose this title. So Klaus and I, in 2012, started a joint venture between the CNRS. So I belong to my employer. The people who pay me are in France, the CNRS. And we started in 2012 a joint venture between the CNRS and the University of Illinois. And so Klaus came to visit me in Nancy. So for those who don't know Nancy, this is the capital of Lorraine, in the northeast of France. And this picture was actually taken during a little press conference that we had. And so we had like a bunch of journalists asking questions, and at some point one of the most clever journalists asked Klaus, what kind of interest do you have? You're a very well-established professor, and you come from this great university, 25th in the Shanghai ranking, and what kind of interest do you have to collaborate with someone from this 300th university in the Shanghai ranking? And Klaus poked at my head and he said, I have the muscle, he has the brain. And that kind of illustrates what Klaus Schulten was about. Always having a nice word to say, which kind of, is in line with his proverbial generosity and his communicative enthusiasm that we've heard these past 2 days. I don't think that the journalist, incidentally, I don't think the journalist believed that word that he said. Anyway, so I've been interacting with Klaus for 15 years, actually a little bit more. And as we heard today, Klaus gave a lot of freedom to the people he worked with, and so he invited me, never imposed anything, invited me to work on different projects and with different people who, over the long run became, more than just collaborators. I can proudly say that they are my friends. And I'd like to talk about two things today. The two last projects that I had with Klaus. One is on membrane permeability, and the other one is on ATPase. So let me start with the membrane permeability projects, and the people who are involved in this project are, of course, Klaus and Jeff Comer, who is also part of the family. He would be kind of the grandson of Klaus because he was the graduate student of Alek Aksimentiev. And then on the right-hand side, also people from the Schulten family. I wanted to put them here because they're not really involved in what I'm going to talk about, but we were part of a community project that was triggered by Rommie Amaro and JC Gumbart, and so we worked. We tried to figure out what are the best methods to calculate these permeabilities. So there is an impetus for working on permeabilities, and this impetus comes from pharmaceutical industry, and one of the tenets about permeability and bioavailability is that there's a very small percentage of molecules entering clinical trials that will actually make it to the market. And if you think about anti-cancer drugs, 95% of the molecules that are tested in clinical phase 1 are rejected and they will never see the light on the market. And pharmacokinetics is part of the problem. It's not all the problem, it's part of the problem. I was mentioning anti-cancer, but it's the same thing for Alzheimer's disease. And so it's been kind of a holy grail in pharmaceutical industry to predict upstream from this expensive synthesis and clinical trials, try to predict molecules that can actually pass the natural barriers, especially from the gastrointestinal tract, and go into the systemic circulation. But things are a little bit more complicated than they seem. So the first thing, I want to emphasize that I'm only talking about drugs, oral drugs, that are transported in a passive fashion, so not what we heard yesterday from Emma. These drugs don't involve specific transporters. We're talking about passive passive permeation here. So from the moment the drug is ingested and then dissolved in the stomach, and so then it's in principle it will go it will permeate the GI tract and then arrive at the liver in the portal vein, in the hepatic vein, and subsequently go into the systemic circulation. Now, at the level of the pharmaceutical industry, they're using different kinds of assays, in vitro assays. I mean, they do also in vivo assays, but they also like to use this in vitro. I mean, of course, you cannot test all the time in vivo, so they have different models to actually predict bioavailability. And these models use different kinds of cell lines. I mean, the most common, most popular are the colorectal carcinoma cells, which are apparently good models of the GI mucoses. But there are new competitors, canine kidney cells, which apparently are easier to produce and that's very important, have a lower level of transporter expression. But when you talk with people from pharmaceutical industry, they always say that there is no real universal model that they're completely happy with, and which makes our life pretty difficult. I mean, if you want to if you want to predict bioavailability based on calculations, I mean, what should we base our results? What should we compare our results with? And it's kind of a complicated exercise in pharmaceutical industry, because I mean, you have all the time this kind of cycle. You do this in vitro assays, and you find out that the drug is not really good in terms of bioavailability, in terms of PK. So you go back to the synthesis and try to optimize the bioavailability, but at the same time, you don't want to destroy the KD, you don't want to destroy the binding affinity for the target. So these are the typical plot that we get, and I'm going to go in a minute. I'm going to explain how we obtain these profiles. Those are free energy profiles, basically the reversible work that is needed to bring this molecule across the lipid bilayer. And so these simulations have been done in POPC, which is pretty far from, as you can imagine, from the reality of the GI tract membrane. And so Z equals 0 is the center of the bilayer, and minus 40 plus 40 is, you are in water above and below the bilayer. But really, I mean, what should we use as a model? I mean, if we want to make this calculation useful to the pharmaceutical industry, I mean, what should we use as a model? And I want to impress upon you that if you want to make your model a little bit more complicated than POPC, and you add cholesterol at a ratio of 30%, then you can see that your fringy profile, your fringy landscape is already morphed by the presence of this additive, the presence of the sterile molecules. And I can assure you that these calculations have been done very carefully when we're talking about multi-microsecond simulations. So, what's the theoretical framework to do these calculations? This is the kind of assay that we're using. So we have lipid bilayer, we have the drug molecule, and we rely on this model that is called the solidity diffusion model of permeation, which relates the resistance against permeation to the exponential of potential of mean force and the diffusivity. And so what we do is we use an ad hoc representation of the reaction coordinates, and in this case we just use Z, but you can already imagine that this is a very coarse variable that we're using. I mean, it doesn't take into account the relaxation of the lipids as the drug permeates. I want to, going back to Klaus and the second ingredient of the solidity diffusion model, which is the diffusivity, I want to go back to a study that we did that was published in 2008. We revisited a paper that was published by Klaus and Amaro back in 2001, and that was also, there's also something to say about Klaus. I mean, a lot of people kind of consider whatever they published as carved in stone, and you don't go back. You don't. But Klaus somehow wasn't completely confident about the results that he had published in PNAS, no less. But the potential mean force that he had published for the passage of this visceral molecule in the channel was very noisy and kind of reflecting the non-equilibrium nature of the calculation that he was doing. So when we had put the adaptive biasing force algorithm in NAMD, we endeavored with his blessing and with his help to revisit the problem. And at some point we wanted to build a kinetic model, and then we were faced with the question, how do we calculate this position-dependent diffusivity? And then we used, so Klaus had the intuition that we could, somehow use the biasing force to get the diffusivity, but at that time we in spite of spending hours on the board, and we were not able to figure it out, and we basically turned to a paper that Gerhard had published in 2005 relating, actually also using a theory that was also cooked up by Benoit in 1994, relating the diffusivity to the variance of the position. But and then of course we use this diffusivity and the potential of mean force to calculate the mean first passage time, and basically build a kinetic model to rationalize the translocation of glycerol in GLPF. Had to wait until 2013 and the talent of Jeff Kohler to actually utilize the results of a simulation by an adaptive biasing force simulation, so this is biased simulation. And the way we do it is, so we have the actual trajectory, and that's also kind of takes on the same paper by Gerhard in 2005, in which he had put like the seminal ideas to use Bayesian inferences to get to diffusivity. So the idea here is that we have the adaptive biasing force simulation, and we get the trajectory of the collective variable, the coarse variable, and the biasing force. And we pick some trial parameters, which are the potential of mean force and the diffusivity, although we don't need to take the PMF, we already have the PMF. And we assume some propagators, so the Brownian dynamics, which is embodied here with the drift term and the variance term. We calculate the probability of the trajectory, the given parameters, and then we do kind of the inverse solve the Smolikovsky equation, and so we try to get the probability of the parameters given the trajectory. So that's what we do, and of course for a very simple system like looking at the diffusion of water in water with a position-dependent thermostat, which you can implement with great forces. So basically change the friction at a certain position in the box. Then we obtain a pretty good consensus in the diffusivity profile over a pretty wide range of lag times, so that's pretty good. So now we going back to the work that we did with Klaus. Our first application for the calculation of probability was looking at the translocation of water, and one thing that we saw is that you have to use big patches, because even something as small as water will induce a big perturbation in the bilayer, or undulation in the bilayer, which cannot be accommodated if your patch of the membrane is too small. Also, what we observed and kind of lesson to be learned from this simulation. You need to go to long simulation actually to converge the diffusivity. And over 2 microseconds, we're able to reproduce almost the experimental permeability that was estimated by the group of John Nagel. But what we also saw is that there may be it's kind of a result, the fortuitous cancellation of errors, because we're using TIP3P, which notoriously overestimates the diffusivity, the cell diffusion, by a factor of 2, but it also overestimates the subsolvation, the hydration-free energy. So we're making an error here, a little one. We're making a bigger error here, but by virtue of a ratio, an exponential over the diffusivity, and we kind of compensate these errors. So these simulations are pretty expensive, and we thought that maybe we could actually extend the theoretical framework by introducing an extra degree of freedom, because when the permeability goes from one side of the bilayer to the other side of the bilayer, it has to reorient. And so we extended the framework by, of course, revisiting Klaus' lecture notes. And I mean, you know, this is like a treasure. I don't know what's going to happen to these lecture notes, but someone has to do something about it, and they should be published in some form. Because I mean, there's no textbook out there, you know, available to the public that covers as much that is covered in these lecture notes. Anyway, so using these lecture notes, and we actually calculated the not only the translational diffusivity, but also the orientational diffusivity, and we saw that there's a strong dependence of free energy and orientational diffusivity when the permeability goes from one side of the bilayer to the other side of the bilayer. It has to reorient. Now, one thing that we didn't appreciate is that that for alcohols, for instance, we noticed that for this particular course variable that we're using, and I'm not saying how much we can claim that it's going to happen for a complete, for the true reaction coordinate, but a very simple, very naive reaction coordinate model that we're using. There is no consensus on the lag time for the diffusivity. So you see that we have strong dependence of the diffusivity on the lag time. And then what's the correct diffusivity? That's not so easy to answer. And so if you consider the diffusivity as a function of the lag time, a different position in the bilayer, so when we are in the water, in the aqueous medium, and you see that it's pretty flat, but then when we are below the head group region, then we see this very strong dependence of the diffusivity, which is kind of a hallmark of non-Markovian dynamics. And actually, if so you run long series of independent simulation, we run 3,000 independent simulations, 1 nanosecond each, and we obtain this profile, which gives us the probability to go from position Z1 at time T1 to position Z2 at time T2. We see this cusp-shaped curve when we are below the head group region, but a more Gaussian distribution is expected when we are in water. We have this superimposition of 2 different regimes. We have long waves of the nearly immobile substrate, you can see here, and then we have very rare long jumps, which you can see here in this movie. What you see here in purple are voids that are created in the bilayer. So as you see the typical relaxation of the bilayer, things are moving very slowly, and then the liquid chains, when they are dancing around, they're creating voids And so the permanent is actually rattling around and it doesn't move much. But then when a void forms, then it kind of teleports itself in the void, pretty much like a levy flight. And so for this particular, again, I want to emphasize for this particular coarse variable that we use, is a very coarse variable that we're using. We cannot use Gaussian statistics, we cannot use a Markovian description, and so we turn to a different picture. We turn to fractional dynamics, and so to the equivalent of the Small-Gauss equation, this fractional Small-Gauss equation, which gives us a fractional order. So you can see that this fractional order is almost close to 1 when we are in water, and then it can go as low as 0.65 when we are in the middle of a bilayer. And so we have also a fractional linearity, which has some very strange units. So it's angstrom squared per nanosecond to the fractional order. But then, you can see, lo and behold, now we don't have any dependence on the lifetime. So we apply that to a series of alcohols, and we were able to describe the permeability of these alcohols, and we were also able to actually bring to light a flawed interpretation of experimental measurements of the permeability, which are indirect measurements that are actually, nobody noticed that. I mean, it was kind of unnoticed when the publication came out. This permeabilities are nearly 3 orders of magnitude lower than that of water, which doesn't make sense. Now, a little historical intermission. So, it was this dialogue between Bonaparte and Laplace. And so Bonaparte was surprised when Laplace published his treatise on celestial mechanics. Was surprised that Laplace didn't refer to the creator, which is kind of the Occam razor hypothesis. And Laplace said, I didn't need it. And as a scholar, I must provide you with works permitting predictions. So we want to do predictions. I mean, at some point, we would like to be predictive for the pharmaceutical industry. So this is what we're heading for. So we are now at the point where we can simulate more complex membranes, mammalian membranes. So right now this is an ongoing effort with Yi Wang in Hong Kong on simulating a mammalian membrane with 24 components. But what we also would like to do is make this calculation a little bit cheaper. I mean, in pharmaceutical industry, they're not going to simulate for 4 microseconds to get a potential mean force. And what we realized with Jeff Comer is that if you just consider 5 critical points in the potential mean force, you can actually use some cubic splines to actually describe the complete free energy landscape, and then you can maybe turn to perturbation theory to actually just calculate a few points, these 5 critical points, and then fit this cubic splines to model the free energy landscape that you can use subsequently to infer the permeability. Einstein, go ahead. I have 5 minutes to talk about ATP synthase. I will be very short. I will be extremely short. Well, first, I would like to. So I would like to mention the people who actually did the work, and the architect of the project was definitely Alek. It's also Martin Muradi. For the confirmation transition, and John Wildenstein and Takeshi Murata for the structures. I'm not gonna, you've seen that it's cute. I mean, you've seen that in Neil Hunter's talk. Basically, nature evolved a very sophisticated machinery to make bacteria that live in most inhospitable environments survive and survive with only a few photons. And so you've seen this already. So I have to skip. I mean, if I only have 5 minutes, it's no way I can show you this movie. What I want to show is actually the object on which we worked, and so this is kind of the last object that Klaus was really, eager to work on, and he had been working on 10 years ago. Actually, Gurvich mentioned that work, but we now have the tools to actually tackle the problem and understand how the chemical energy is transduced into mechanical work. So we worked on the V-type ATPase, which we use is kind of, it's much simpler than the F-type. You can see here the number of revolutions as a function of time. For V-type, there is no dwell time, whereas for F-type you have this dwell times. V-type actually, V-ATPase work as ATPases, and they basically convert ATP into ADP plus P. Although there are species, it was shown by the group of John Rubinstein for Thermus thermophilus, it can also work in the synthase mode. And so this process, the torque that is created is propagated to the VO part, which is located in the bilayer. So what we did with Klaus and Abi and Mahmoud is to devise a workflow which is sketched here. So on the left-hand side you have the fuel source, basically the origin of the chemical energy, and on the right-hand side you have the conformational transition. And so basically what we are after is understand how the energy that is produced in the hydrolysis of ATP is transduced into the A3B through the central stop into mechanical work. So for that, we had two different assays with featuring Enterococcus aureus. One assay bereft of the central stalk, and one assay with the central stalk. The reason why we did that is because the group of Murat actually showed that ATPase bereft of the central stalk is still capable of fulfilling enzymatic activity. So we used a collection of methods, starting with an isotopic network modeling, target MD, string method with form the trajectory, Bayesian sampling, into multicolor MD to get the diffusivities in the spirit of Bayesian inferences. So let's start with the chemical work, and I'm not going to go to the details of the different states that we'll be looking at, but suffice to say that we considered a number of possibilities corresponding to the different states of different conformational states of the A3B3 ring. So it works in ATPase, and like ATP synthase, it works in true 120-degree catalytic cycles. And so you're going from an empty state to a bindable state, to a bound state, to a tightly bound state. And so the way it works is, so when the products are ejected, now we go to an empty state, now we have here a bindable state, and ATP goes in, and then here we had ATP in bound state, and it goes into a tightly bound state. And so to get access to the chemical energy, so we did this free energy perturbation calculations although we did it done also was like a geometrical transformation, potential reinforcement calculation. And we used an approach that we cooked up with Brunger and J.C. Gumbart. The philosophy here is when you do these binding affinity calculations, so basically decouple the substrate from the binding pocket, then the risk, of course, is that the substrate goes away as you decouple it. And when you want to do the reverse transformation, then chances are that it will never find back the binding pocket. And therefore you kind of violate thermodynamic microreversibility. So the philosophy that we have is that if we cannot simulate it, then restrain it. So for that we introduce some groups of atoms in the protein and also in the substrate to define the degrees of freedom that allow us to define the position and the orientation of the substrate with respect to the protein through the 3 order angles and through the 3 spherical coordinates. Now, any restraint that you add to the system that corresponds to a loss of configurational entropy, which you need to take into account when you calculate your binding free energy. And this is embodied here in ratios of configurational integrals. So we have a configurational integral for each degree of freedom. And of course we have to do this decoupling both in the bound state, but also in the unbound state. And for the unbound state, there are some terms that you can calculate analytically, typically the tumbling, the translation of the substrate in a homogeneous environment. Anyway, so that being said, we have for the different states that are available to the A3B3 V1 ring, we have done these calculations, which are pretty expensive to do, and we find that the chemical energy that is produced in the hydrolysis of ATP corresponds to about 7.5 kcal/mol. So now what do we do with this 7.5 kcal/mol? So we return to the workflow, and now we'll see now how this energy is transduced into mechanical energy. Now we will consider the conformational transition that is involved in the catalytic cycle. And the way we do that is we start with the crystallographic structure that is available. We need to construct different models with the nucleotides, different types of nucleotides in the binding pockets, and then we generate a guessed string using very coarse approach, targeted ND, and we go to a swarm of trajectories. So you already heard about swarm of trajectories, so I'm not going to give you the detail of that, but as Gerald was saying, we're not doing dialanine anymore, but it's always nice to see something in 2 dimensions for a string, because in the case of the A3B3 ring, we're actually working in a dimension of almost like 1200, which is more difficult to visualize. But the idea is still to get a string, to get a pathway, a minimum free energy pathway. We're not making any claim here that is the minimum free energy pathway. Actually, in the case of dialanine, starting from this guess string, we are able to identify one possible pathway, as was shown by David Chandler in 2000. Actually, 3 different free energy and minimum free energy pathway by virtue of the periodicity of the Chandler angle. So now we have the string, and with the string we can do the free energy calculation following the string. So the strings are formed by images which correspond, we can visualize that as snapshots of the A3B3 ring, going from one catalytic state to the next catalytic state 120 degrees later. And so we obtain this potential of mean force. What you have in orange is the potential of mean force of the V1 ring devoid of the central stalk, and what you have in red is the potential of mean force with the stalk. As you can see, the presence of the stalk lifts the degeneracy of state 3, and we have two different states here. We have the bindable state and we have the bound state, which incidentally have been shown by the group of Murata to be actually to exist. I mean, the intermediates that were predicted by these calculations, and the string calculations actually have some basis in fact. So I went too fast. I'm sorry. But what you saw in the previous slides is that you're gaining 6 kcal/mol, and the potential of mean force, the fraternity difference, was 6 kcal/mol. So back of the envelope calculation, the efficiency of the energy conversion would be about 6 over 7.5. So 7.5 comes from the chemical work of the hydrolysis of ATP. 6 is actually the mechanical work that was produced. So 6 over 7.5 will give you about 80%, 75%, 80% efficiency of ATP. So we use the Bayesian inference to get the diffusivity, and from the diffusivity we got mean first passage times, and the inverse of the mean first passage times is the rate constants. So we have the rate constants for the different steps in the conformational transition. If we look at the trajectory, and we learn a lot from this long trajectory. We learn that there's a lot of concerted motion when we use ZANS, the tool developed in the ZANS group for allosteric motion analysis. We saw that we have a concomitant swiveling motion, sliding motion of the A and B subunits, which actually induce, so there is definitely a causality relationship. You have this motion, swiveling motion, sliding motion, which induce the motion of the stalk, and all of this is facilitated by water, which acts as a lubricant. I'm sorry, I really have to. So I'm almost done. So the conclusions here is the work that we did with Klaus, with Abi, with Mahmoud definitely eliminates a thermodynamically favorable pathway with a clear causality relationship of going from ATP undocking to ATP binding to stalk rotation to ATP hydrolysis. Most of the energy that is required for the rotation of the stalk is harnessed within the AB protein interfaces of the V1 ring, and the mechanical properties. Actually, I didn't mention that, but we also did like an elastic modulus analysis of the stalk. What we saw is that the mechanical properties of the stalk are perfectly tuned to optimize the kinetic barriers of rotation, and hence enable millisecond rotation times 2. So where are we heading for? We're not completely satisfied by the 80% efficiency of the mechanical transduction. In nature, we know that it's probably closer to 100%, and we kind of ascribe that beyond the usual culprits, the force field, the insufficient sampling. We also ascribe that maybe to the absence of the VO domain, which could add some rigidity to the motion of the stalk. So this is something that we are currently exploring. Another thing that we are exploring is the effect of spurious mutations on the functioning of ATPase. Again, the group of Murata showed that there are some mutations that actually loosen the interaction between the A3B3 ring and the star. And there are some mutations that conversely actually rigidify this interaction or reinforce this interaction, which of course has some bearing on the rate constants in the function of ATP. I'd like to end with a picture that is really dear to my heart. It's a picture of 3 generations of Beckman Fellows, starting with the youngest, with Angela Barragan, who is actually a PhD student of the TCBG and currently in Odense working with Ilya Solovyov. She is a young Beckman Fellow, graduate Beckman Fellow. Abhishek was a postdoctoral fellow, and I was a senior Beckman Fellow. All of this to say that I'm deeply indebted to the Beckman Institute for facilitating my visits whenever I came here. I also want to say that one thing about Klaus, that Klaus really wove a family, and I'm really proud to be part of this family. With that, I would like to thank you for your attention and your patience.
So now we have closing remarks by the friend and the neighbor. Can you write it?
Well, it's an honour and a pleasure to have been invited to speak at the symposium. Although I was actually a colleague of Klaus in the physics department for nearly 30 years, our academic interests didn't on the whole overlap very much. I think, in fact, perhaps the biggest contributions that I made to Klaus's work is probably that I encouraged my PhD student, Johan Kostin, to transfer to his group and start working on biophysics. Johan had a very successful career in that group. He's now moved on to the University of Missouri in Columbia and actually got the DeGen Prize a few years ago. But as Alexa has mentioned, I actually knew Klaus far better as a neighbour, a next-door neighbour on Pennsylvania Avenue in Urbana. We moved, my wife Haruka and I moved to Urbana in 1983, and a few years later, Klaus and Zan arrived and took the house next door. And I think I can say with some confidence that there was at least one group of people in Urbana to whom Klaus and Zan's occupation of that house brought a great sigh of relief, and that was the Urbana Fire Brigade, because the former tenant was an old lady who lived by herself and had a passion for collecting old newspapers. And as a result, the 6009 West Pennsylvania Avenue was the worst fire hazard in the whole of the city of Urbana. Zan and Klaus probably prevented that. Well, they enjoyed them as next-door neighbours for nearly 30 years now, and I think you've heard of some of Klaus's talents and interests. So you've heard about his cycling, perhaps you've heard about his berries, his enthusiasm for berries. One thing you might not have heard of was that he was in fact took an enormous interest in technology. In that respect, although we were colleagues in the Department of Physics, he and I were polar opposites, since I am the sort of paradigmatic E-class. I have not yet caught up with the information revolution. I suspect I never will. Anyway, so we were really at two poles, as it were, the opposite end of the spectrum. Luckily, we didn't mutually annihilate, and in fact, Klaus and Zan were very helpful to us, and both our coffee machine and our television owe a lot to But I did have one fairly brief episode in which my academic interest did intersect rather closely with Klaus's, and that was in the context of his work on electron transfer in proteins this was in the 90s, actually. Now I should acknowledge that I'm probably speaking in the presence of a large fraction of the world's top experts on electron transfer in proteins. I am not a computational biophysicist. I'm not even really in the open quantum systems business. So you'll have to forgive me if I deal very lightly with both the prehistory and the post-history of this interaction. But I would like just to explain how it worked. So, Klaus was interested in the early mid-90s in a bacterium, I guess, known as Pseudomonas viridis. I guess all of you probably know this piece much better than I do. I've never been introduced to it, but here is my naive condensed matter physicist cartoon of what goes on in electron transfer in this beast. So what was observed, what had been observed experimentally, was that the backward rate at room temperature is very slow. The forward rate at room temperature is about 3 inverse picoseconds. What I think caught Klaus's interest was that it increases by a factor of the order of 4 if you go down to 8 Kelvin. And generally speaking, that's, I believe, usually taken to be an indication that quantum mechanical effects may be important. It was also noticed that there was not much dependence apparently on the redox energy. And again, that was a bit of a puzzle. So, if we look at it from the quantum mechanical point of view, this is a special case, a more general two-state problem. You have some object system, perhaps an electron, perhaps something different, which is trying to hop from a left well, say, to a right well. It has to traverse a potential barrier, and it presumably does that by tunneling. In the case of present interest, the environment is the vibrating nuclei of the protein, and of course that means, as you all know, that the nuclear coordinates are coupled to the tunneling electron by the Coulomb force. In the most general version of this problem, presumably we would need to consider both the effect of the coupling to the environment on the actual barrier transmission, and also the effect of its coupling in detuning the initial and final states. Now, apparently, in the present case, the people, I believe, tend to think that the Born-Oppenheimer approximation is probably good, and therefore you can neglect the first effect and just concentrate on the detuning effects of the nuclear motion. And what Klaus recognised was that this then effectively makes the problem a sort of rather classic problem that many people in the open quantum systems community had been interested in, which is the so-called spin boson problem. So let me just say a brief word about that. In the generic spin boson model, you have a system, which is your primary interest. You have some kind of fluctuating environment, and you have a coupling trigger. And this can be put into correspondence with the problem of a spin of a half interacting with a complex environment. And the important parameters are the Baire tunneling matrix element, delta, and the offset, epsilon. And so in the open quantum systems community, one had basically thought of this in terms of the diagram here, and written down a specimen form of the environment Hamiltonian and the coupling. Now, you'll see that the environment Hamiltonian actually describes a set of simple harmonic oscillators. The question of when you can and can't use that description as a set of simple harmonic oscillators is actually a quite tricky one. We thought of it in some contexts, but I'm sure some of you thought of it in other contexts. But at least for relatively simple environments, very often it does seem to work. The coupling term, again, is a rather simple form, it's simply linear in the oscillators. In this case, essentially, what we can think of this, I think, as a multi-mode model, and I've actually indicated the correspondences between the chemical physics problem and the spin boson model as we've been thinking about it in these correspondences here. And what presumably we'd like to know is the transfer rate as a function of the redox energy, epsilon, and temperature, T. Well, it just happened that we, coming from a totally different background, a different point of view, we were actually interested in the possibility of displaying quantum mechanical effects at a level approaching the macroscopic level. Well, we had become interested in the spin boson problem, and a group of us had actually got together to think about it. And it turns out that if you are interested in only, So we've been thinking about it. So Klaus and I and some of our students got together and tried to think about the correspondence here. And it turns out there's a very critical feature of the spin boson problem. Provided we're only interested in dynamics of spin, then we can get complete information about the effects of the environment from this one function, the coupling spectral density, J over omega. And that can often be obtained from classical arguments. For example, in superconducting devices, which what we're going to be primarily interested in, we actually could try to derive the spectral coupling from the behaviour in the classical regime. Klaus realised that he could actually get the information needed about this quantity J of omega from a classical molecular dynamics simulation. Well, I've just written a few words there. Obviously, the work involved in actually extracting the J of omega from the computation was highly non-trivial indeed. And this was a large part of the content of the papers I mentioned at the beginning. What they did find was that at least an approximate good form of J of omega is given by the expression alpha omega over 1 plus omega squared choice squared. Now, if you, just for the moment, ignore the omega tau in the denominator, you set that much less than 1, then what that means is that you have what's called, what in the open quantum systems community is called ohmic dissipation, and a crucial quantity which controls the behaviour in the presence of ohmic dissipation is the quantity, the alpha which sits in that expression. And it turns out that you can actually work out the numbers. I've actually tabulated some of them there. Perhaps the most important point is that alpha, the dimensionless number, is very rather large compared to 1, something like 25. This means, in effect, that our system is an ohmic, a strongly overdamped ohmic system. Well, we have actually been interested in some sense the more general treatment of the spin boson problem, and a group of us, half a dozen of us, had actually written a very lengthy paper in Reviews of Modern Physics on this. I should say this paper is something like 80 pages long. It is we had 6 authors, and for 5 of them, it is our most highly cited paper of our careers. I should also say I regard it as the most boring paper but basically, we did try to address the spin boson problem in as much generality as we could. And the formulation we gave, which is perhaps not quite the same as the one that's appropriate in biophysical problems, and given the form of J omega, and of course given the parameters, the free system parameters, delta, epsilon, and t, basically you set the initial condition to be +1 and simply evolve according to the total Hamiltonian. You calculate sigma z, and if it's exponentially decaying, then presumably from that you can extract a transfer rate k. And I assume that does agree with how I normally define it in the chemical physics problem. Well, how we did is not terribly interesting, so let me go quickly through that. It was possible to derive an exact formal expression for this P of t, essentially the preponderance of being on one side rather than the other, in terms of our parameters, and we could represent it in a graphical form. And crudely speaking, the diagrams we drew corresponded to sort of transitions from the diagonal elements of the density matrix to the off-diagonal ones and back again. You have to go into the technical details to understand that, really how that works. But the important point was that under certain conditions, and in fact under a lot of the parameter space, you can justify what we named rather inelegantly the non-interacting blip approximation, which means basically that when the system makes the transition from the diagonal to the off-diagonal elements of the density matrix, it basically forgets about the last time it did it. That's the simple, non-technical way of saying it. There's a lot of technical mess which goes with it. Under certain conditions, it then turns out that you can obtain an analytic expression for P of t, and therefore, when appropriate, for the transition rate. Now, it turns out that in the overdamped case, and only in the overdamped case actually, step 2 gives us the golden rule result, which had, of course, been obtained by other people and by other arguments. We did feel, well, one thing we I think would claim to have done in this long and boring paper was to actually give a rather better justification of the golden rule expression than had previously been given in the literature. So you see, we've got an expression for the transition rate, which is one I'm sure most of you are familiar with, and everything is defined by simple particle parameters, epsilon, delta, and temperature, and by the spectral function, J omega. Now, as I said, we basically studied it in a different context, and we primarily concentrated on the case where J omega was simply a power law for all omega. And we found some moderately interesting results in that case. It turned out that you can evaluate things analytically, and the transfer rate for zero bias, zero redox energy, and non-zero temperature is a power law in the temperature. Similarly, if you go to temperature 0 and non-zero bias, then you get a power law in the redox energy. If they're both non-zero, then you're missed. I mean, you can write it down, but it's not very Now, the interesting thing was that for the current problem in this particular molecule, the cutoff energy, the cutoff frequency, the order of called -1, is quite comparable to the other parameters in problem. And what that means then is that you have to keep the full form of J omega. And in their paper, Klaus Schulten showed that first of all, started off by showing that provided the temperature is high compared to h bar of tau, and so essentially on the classical regime, then the resulting expression can be evaluated analytically. The result is expressed in a form which is essentially a generalization of the original models. This had in fact been noticed by other workers earlier than that. So it's this really formula there. However, in general, what they found was that you have to worry that the fluctuations are quantum fluctuations, and so the expression for the mean square energy fluctuation does have this characteristic quantum factor, cos beta h bar over 2. And lower, so high temperatures, at room temperature, basically they found that the original Marcus result is roughly correct. Lower temperatures, you have to put in quantum fluctuations, and in some cases it's necessary to calculate it numerically. And you have to input specific values of this dimensionless parameter alpha and tau. And in the final figure in the paper, they indicate that at, again, room temperature, Marcus theory works, which is the dashed lines in the figure, works reasonably well. At lower temperatures, there is a rather definite discrepancy, and moreover, the peak gets somewhat asymmetric. So, this part, I should emphasize the business of actually, first of all, actually calculating this function, J omega, is highly non-trivial, and then having got it, the business of finding a reasonable approximation for the evaluation of these complicated intervals and so forth was by no means trivial. So, at the end of the day, what they found was, first of all, again, to repeat, at room temperature, simple Marcus theory works reasonably well. That's not too surprising because room temperature is greater than the other characteristic energy of the problem. At lower temperature, and not going down to zero temperature, the generalized Marcus theory with thermal fluctuations seems to be replaced by thermal plus quantum fluctuations. That seems to work well. And finally, for zero temperature, you need to evaluate the Gaussian expression numerically. And if you are, again, if you're not interested in this particular molecule, but you're interested in a more general problem, for example, liquid solvation, then you have to evaluate the integral numerically, and the odds are you will not get the result which is obtained from a simple theory. So obviously I'm aware, at least, although as I say I haven't followed the literature in detail, I'm aware that there's been lots and lots of work in the subsequent years. We're talking, this is in the 90s, remember, and over the last 20 years I know there's been lots of work. In fact, I'm conscious that my subject of this talk was certainly related very strongly to what Dr. Klein-Cutter talked about. I suspect it's also related to what Dr. Hammer Schiffer talked about, though unfortunately I couldn't hear her talk. But I think this work of Klaus was perhaps the main point was that it perhaps showed, I don't know if it's perhaps for the first time, but it certainly showed that if you were energetic enough and were prepared to do molecular dynamics real hard, molecular dynamics calculation, then you at least could plausibly get out the transfer rates for a real physical system and not just for some kind of abstract model. Remember, this is a very definite model we're talking about, which I wrote about. As I haven't followed the developments of this work in the last 20 years, but I would like to just throw out one question, and I don't know if people are already discussing this. As many of you know, right now the prospect for making a general quantum computer which can beat out the best classical one and not that that's not that optimistic. On the other hand, there are a class of special problems which you can build a special purpose quantum computer, and the prospects that may beat out the best classical one are a lot better. And specifically, there's been a proposal a couple of years ago now, I think, from the music group at Harvard that so-called Bose sampling technique is sort of very scaled down kind of quantum computer, but that actually might be helpful in, for example, investigating electronic transitions in funding, and might within a few years actually beat out a classical computer on that. So I'm wondering, as far as I understand it, the problem of electronic excitation in funding is not that qualitatively different from the problem that Klaus was looking at. So the question is, have people thought about this? Would boson sampling in fact help with this general program and with the related problems? Well, I guess we perhaps some of you know the answer to that question. But whatever the answer is, I think that Klaus, looking down from wherever he is, would be very interested in this, and wishing you every success in your continuing the work he's doing. Thank you.